representing different schools of teaching disciplines. So we all have seen that from last 2 years, pandemic really hit the world in a very drastic manner and turned all the tables. So even as of now, we do not know how this year will unfold, and we continue to live in all the aspects of our lives with more than the usual question of trepidation and uncertainty. But no matter how many colleges and universities had been shut down, how many examinations have been postponed, IRI celebrated and validated the graduation ceremony even in the last year, that is 59th convocation, by following proper rules, guidelines, and adopted every change which was the need of the hour. So this time also we have come up by overcoming all the limitations and technological constraints from our last year experience and along with the better streamlined flow. I hope you all have witnessed the same things in the last 2 days, that how perfectly our program was done. So yet I hope you feel, as I do, the energy of this moment and the anticipation and the excitement of our convocation week. So without any delay, I request our Honorable Dean and Joint Director Education, Dr. Rashmi Agarwal, for welcome address for today's event. Dr. Rashmi Agarwal.
Thank you, Simardeep, with a very nice introduction. Good morning to all of you, and it is my pleasure and proud privilege to welcome all on the third day of our 60th Convocation Week program. So today we have significant achievement presentation, educational achievements by the professors of various teaching disciplines at IRI. So in the forenoon, we have 3 sessions. First session pertaining to the School of Crop Improvement, second on the School of Crop Protection, and the third is on the School of NRM. So the significant educational achievements for the year 2021 by the professors of teaching disciplines of the Institute, this will be presented by the professors, and the convener is for these sessions Dr. S.P. Datta, Professor, Soil Science and Agricultural Chemistry. So on this occasion, I very warmly welcome all of you. First of all, I would like to welcome our chairman of the first session, that is School of Crop Improvement. The chairman is Dr. Kuldeep Singh, former director NBPGR, and at present he is cluster leader, Gene Bank, and head consultant at ICRISAT, New Delhi. So we all of us warmly welcome Dr. Kuldeep Singh, sir, on behalf of all the faculty, on the behalf of director, faculty, staff, students, and on my own behalf, I warmly welcome you, sir, on this first session of the third day of the 60th Convocation Week program. And you will be chairing the session on the School of Crop Improvement. These professors of 3 disciplines will be presenting the educational achievements of the students carried by these disciplines. These are our genetics and plant breeding, plant genetic resources, and seed science and technology. And there are total 39 students who will be getting the degrees during this convocation, and the significant achievements will be presented by the respective professors of these 39 students. So let me share with you that our research activities and educational activities at IRI constitute a very important part of Indian Agriculture Research Institute in total achievements. And during the year 2021, a total of more than 300 publications have emanated from the students' thesis themselves. And it's really heartening to know that more than 75% are the NAS rating more than 6. So you can just imagine how much significant work our students are carrying out. And the testimony of it was that yesterday and day before we had the significant achievements by the students for the merit medal and the best student of the year award. Now this we also have converted into NABARD Professor V.L. Chopra best student of the year. So with this brief background and introduction, I would like to welcome once again each one of you, all the professors, head of the division, and all the faculty, students, staff, ladies and gentlemen. Once again, all of you are welcome. And I profusely welcome our Director, Dr. Aki Singh, and Vice Chancellor IRI, who is a pillar behind all these activities and the guiding force. So, sir, I welcome you also along with all the faculty members. And Dr. Kuldeep Singh, you are profusely welcome. And I would like to now request Dr. S.P. Datta, who is the convener of to take up the further proceedings.
No, Simardeep will continue, inviting the chapter director.
Okay, so
Simardeep.
thank you, ma'am. Thank you so much. As you have rightly said, I also believe that past few years have involved some of the most memorable and influential moments of all the degree recipients. They have really worked hard, and we have witnessed clearly in the last 2 days. Individually, their experiences are very unique here, but all together they share a common bond. So with these words, I'm honored to have the opportunity to invite our Director IRI, Dr. Aki Singh, to introduce or to welcome the chairperson and to say a few words to initiate the event. Dr. Aki Singh.
Thank you, Simran, and very good morning to each one of you. It's my proud privilege to introduce to the audience the distinguished chairperson of this particular session on crop improvement, Dr. Kuldeep Singh, a great colleague of mine. Dr. Kuldeep Singh did his master's and PhD from Punjab Agriculture University, Ludhiana. He served that university for a very long period as wheat breeder, geneticist, molecular geneticist, senior molecular geneticist, director of a School of Biotechnology. And as a postdoctoral fellow at International Rice Research Institute, Dr. Kuldeep Singh did pioneering work of developing a complete series of trisomics in rice, the work which is most acclaimed and internationally cited. And it has been used subsequently, intensively in understanding the rice genome after it was sequenced. Also, Dr. Kuldeep Singh has been part of the International Consortia on Wheat Genome Sequencing. And he was deeply involved in sequencing chromosome 2 of 2A of wheat. And this paper got excellent international recognition with publication in journal Science. His contribution to wheat breeding and particularly on wild hybridization, bringing genes from wild relatives to cultivated species, both in case of rice and wheat, is remarkable. One of the genes, XA38, which he transferred from Raija nivara, characterized, mapped, and fine mapped, and shared with the entire breeding community in the country. And we were also the beneficiary. We used that gene in our breeding program and transferred that to Pusa Basmati 1121 and released a improved version of this called Pusa Basmati 1718 with resistance to bacterial leaf blight. It was a great partnership. Also, we had a number of other projects where we did work in association. One project was on tilling on Nagina 22. He is an excellent teacher, and his role in capacity building, human resource development is phenomenal in field of biotechnology. He also worked as director of National Bureau of Plant Genetic Resources, and that's where he played a key role in initiating mega projects, at least 5-6 mega projects for characterization of germplasm that is locked in gene bank. And these projects cost something like more than 200-300 crores total put together, including rice, wheat, millets, and oilseed crops. So this was a great initiative by Dr. Kuldeep Singh. Also, he took out many of the germplasm lines for their phenotypic characterization in large area. And so this has been a great contribution of Dr. Kuldeep Singh, and truly recognizing, his contribution in genetic resources, plant genetic resources. Hardly he got time to rest even a single day. The day he superannuated from his job from NVPGR, he was requested by CRISAT to steer their gene bank program. And that's where he is working. And just a few days back, we saw him, interacting with the Honorable Prime Minister, explaining about the CRISAT activity and its gene bank. It's indeed my great pleasure having great association with Dr. Kuldeep Singh and to welcome him to chair this session where the significant research finding of the students in the crop improvement is going to be presented. The one student selected from each discipline by a divisional level competition, they have presented their significant research finding yesterday and day before yesterday. for competing for the gold medal, but the students who could not be part of that presentation, their work is no less significant, and that will be presented by the professors of the respective discipline. So with that, I would like to once again extend warm welcome to Dr. Kuldeep Singh to chair this session. Thank you.
Thank you so much, sir. And I express my warm welcome to Dr. Kuldeep Singh, chairperson for today's event. And it's always been a great experience to learn from you, sir. And we are looking forward to hear from you today also. So with these words, I'm pleased to invite Dr. S.P. Datta, Professor Soil Science and Agricultural Chemistry and convener of today's event, to brief us about today's sessions and to conduct the further proceedings. Dr. S.P. Datta, thank you.
Yeah, thank you very much, Simran. Honorable Chairman of the session, respected Director IRI, Joint Director Education, Research, Extension, Associate Dean, Heads, Professor, Scientists, dear students, dignitaries, ladies and gentlemen. It's my immense pleasure to welcome you all to Professor presentation, one of the most important activities of the week-long celebration of our convocation. If we see across the globe, student research, that is the contribution of young mind, have been the baseline of most of the advances made in the science. Hence, this program is very important, where we all will have opportunity to have a glimpse of students' research work across the 26 disciplines of IARI. Unfortunately, we have been undergoing COVID pandemic for last 2 years, but the way our Honorable Director and under his dynamic leadership, Institute handled this situation and provided conducive environment for student research and teaching to possible extent, it is really appreciable, which is also evident in the number of students going to get their degree this year. This is comparable with that of any of the previous normal year. This is indeed a remarkable achievement of the Institute. With this word, coming to now the presentation, the School of Crop Improvement, most important pillar of the Institute, and it comprises 3 disciplines: genetics and plant breeding, plant genetic resources, and seed science and technology. Now, with the kind permission of chair, sir, may we go for first presentation?
Yes, please go ahead. Go ahead.
Thank you, sir. So first presentation will be from genetics and plant breeding, and Professor Binod will make presentation.
Good morning to all. Respected Chairperson of the session, Dr. Kuldeep Singh, Director IRI, Dr. Aki Singh, Dean and Joint Director Education, Dr. Rashmi Agarwal, Convener, Dr. S.P. Datta, Heads, Professor and students. Here I am making a brief presentation of significant educational achievements of postgraduate students of the discipline of genetics and plant breeding. The mandate of the division includes basic and strategic research for genetic enhancement of field crops, applied research of varietal development in various field crops, transfer of technology, postgraduate teaching and research and development of collaborative linkages with national and international organizations. The research work of our students is oriented towards meeting the mandate of the division. We are having 72 faculty members, out of which 48 are located in the division of genetics, and others remaining 24 are located either in sister institutes or at regional stations. This year, total 21 students are eligible for getting their degree. 13 students are getting their MSc degree, and 8 students will be getting their PhD degrees. The broad areas of research I have classified into 4 broad categories: abiotic stress tolerance, biotic stress tolerance, quality traits, and miscellaneous traits. And the crops covered are wheat, rice, maize, mung bean, soybean, chickpea, and pigeon pea. This is the list of students who are getting their MSc or PhD degree. The presentation which I am making here is based on the research work done by these students. Broad area of abiotic stress tolerance. Our students work on drought and aluminum toxicity tolerance in rice, and drought and salinity tolerance in chickpea. PhD student Mr. Joshua worked under the guidance of Dr. Aki Singh. He selected 4 QTLs for imparting drought tolerance in rice variety Pusa 44. He developed monoQTL, diQTL, and tetraQTL near isogenic lines of PUSA44. Standard marker state backcross breeding method was used, and recurrent parent genome recovery was as high as 99%.
Hello. Hello.
Slide is not moving.
Yes, Dr. Burman.
Barman. Hello.
I think
Dr. Barman.
or yes, sir.
No, he will again do it.
I think he is offline or what?
That is a power interruption.
Just check. Barman, क्या हुआ? It's off. The presentation is gone. एक बार जरा उसको फिर से शुरू करिए. कौन? You will have to share again. Who is?
Dr. Amrinder, please unshare. I'll share again. At my end, there may be some problem.
And recurrent parent genome recovery was as high as 99%. Overall, he developed 127 near isogenic lines. Isogenic lines of PUSA44 carrying drought tolerance QTLs were assessed for their agronomic performance under reproductive stage drought stress. Significant delay was observed in days to 50% flowering in PUSA44 and all the nils except those with QDTY 9.1 based which were early flowering. Also, significant yield improvement was observed in nils and it was as high as 2.8 times than PUSA44 under drought stress conditions. PUSA44 QDTY nils were also characterized for grain and cooking quality and were found to be at par with grain and cooking quality parameters of recurrent parent PUSA44 within the treatment. The near isogenic lines developed in this study are ideal candidates for cultivation in drought-prone environments and also useful as donors in rice breeding program. MSc student Mr. Ashish Bhatt worked on aluminum toxicity tolerance in rice. Objectives were to characterize rice germplasms for aluminum toxicity tolerance, marker trait association for aluminum toxicity tolerance, and characterizing allelic variation for NR81 locus. Based on hematoxylin staining and root response pattern, 60 ppm concentration was found to be critically selective for aluminum toxicity tolerance screening. Major findings of the study are listed here. Major phenotypic trait affected by aluminum toxicity is root, and relative reduction in root length on germination was a quick measure to determine aluminum toxicity tolerance. In the germplasm panel, 14% showed tolerance, 4% moderate tolerance, and 82% were sensitive. 8 significant MTA were found in the study, of which 4 were associated to relative reduction in root length, while another 4 were associated to shoot length. Based on the presence of NR81 gene, an allele frequency of 0.11 was found for the tolerant allele. PhD student Mr. Neeraj Kumar worked on QTL mapping for salt stress tolerance in chickpea. He used a real population between
Hello. Dr. Bauerman. Hello. I think some problem is there. Dr. Burman.
I'm calling him.
Oh.
Dr. Amrinder, can you please share it from your end?
Yeah, just I'm sharing from my side.
Good morning to all. The mandate of the division. Good percent. Overall, he developed 127 gas isogenic lines. Isogenic lines of PUSA 44 getting drought tolerance criteria were assessed for their economic performance under reproductive stage drought stress. Significant delay was observed in days to 50% flowering with PUSA 44 and all the males except those with QDTY 9.1 missed, which were early flowering. Also, significant improvement was observed in yields, and it was as high as 2.8 times than PUSA 44 under drought stress condition. 44 QDTY yields were also characterized for bouquet quality and were found to be at par with grain and bouquet quality parameters of frequent.
I think let us first sort out this problem, why it is happening today. Yesterday, absolutely, there was no interruption. आप एक बार देख लीजिए डॉक्टर बर्मन क्या प्रॉब्लम है.
अमरेंद्र सर आप अनम्यूट कीजिए, उसके बाद ही आवाज आएगी.
ppm concentration was found to be critically selective for aluminum toxicity tolerance screening. Major findings of the study are listed here. Major phenotypic trait affected by aluminum toxicity is root, and relative reduction in fruit length on germination was a quick measure to determine aluminum toxicity tolerance. In the germ-plus panel, 14% showed tolerance, 4% and 82% were sensitive. 8 significant MTA were found in the study, of which 4 were associated to relative reduction in root length, while another 4 were associated to shoot length. Based on the presence of NR81 gene, an allele frequency of 0.11 was found for the product. Next student is to on detailed mapping for salt stress tolerance in chickpea. He used a real calculation between the drastic parents, ICCB, and
I think let us first sort out this problem. Amrendra, if your mic is on, then what you are speaking is also heard, and then there is a screen on this. Dr. Vinod.
Factors. Further, through transcriptome analysis, he observed a preparation of MAP, NAC, and Werke transcription factors under salt stress. The tolerant lines ICCV10 and GG11 were found to have high influx of potassium ions and osmotic homeostasis. PhD student Mr. Harish worked on characterization and allele mining for drought tolerance in chickpea. He used 125 land races and 25 varieties of chickpea and evaluated them for 9 morphological and 7 physiological traits under rain-fed and irrigated conditions. He used SNP genotyping for molecular diversity and association analysis. Major findings of the study are genotypes IG5856, PUSA 72, IG5904, IG5849, and ICC 4958 were stable and high yielding. Total 63 significant SNPs were reported for all the traits studied except number of secondary branches. 37 candidate genes were putatively identified from the significantly associated SNPs. Prominent CAP2 gene expression was observed in drought tolerant genotype IG5856, IG5861, and ICC 4958. Coming to biotic stress tolerance, 3 MSc students worked in this area.
Student Mr. Uttarayan Das Gupta worked on mung bean yellow mosaic India virus resistance with the objective to identify key genes and signaling pathways, identification and validation of novel EST SSRs and selected genotypes, and role of phenolics and flavonoids during disease expression. He used contrasting genotypes PMR1 as resistant and Fusa Vishal as susceptible genotypes. Molecular regulatory network underlying MY-MYV resistance was deciphered through RNA-seq analysis. Key genes like MYB and VRKTA transcription factors, LOX and JAZ were identified as the main target for MY-MYV resistance. A set of 513 novel stress-specific EST SSRs were designed and 12 were found polymorphic. Based on biochemical analysis, total phenolic content was found correlated while total flavonoid content uncorrelated with resistance to MY-MIV. MSc student Ms. Purnima Ray worked on inheritance of resistance to mung bean yellow mosaic virus in soybean with the objectives to screen soybean germplasm and to study inheritance of MY-MV resistance. Out of 92 genotypes screened, 7 glycine max and 10 glycine soja derived lines were found to be completely immune. Genetic analysis was undertaken in F2 population derived from the cross SL958 and VLS59 with a total population size of 104 plants in F2. The genetic analysis revealed a single dominant gene for resistance against MY-MV in soybean. She also developed infectious clone of MY-MV, and through sequencing, it was found to be similar with different isolates of MY-MV. Student Mr. Jatin Tamar worked on characterization of dicoccum lines with the objectives to study phenotypic diversity, to evaluate for leaf rust resistance, and to study genetic diversity of selected lines using molecular markers. He used 192 germplasm lines for phenotypic diversity and screening against leaf rust and selected 96 lines for genetic diversity. Out of 192 lines, 19 lines were found to be immune to leaf rust pathotype 77-5. Through molecular analysis, genotypes were grouped into subpopulation. 3 MSc and 2 PhD students worked on quality traits. MSc student Ms. Sharma worked on genetic variability for lipooxygenase activity and validation of LOX3 gene affecting retention of carnyl tocopherol in maize during storage. She used 14 inbred lines, among which 7 are normal and 7 are isogenic lines with favorable VTE4 allele. Self-seed of all these lines was stored at room temperature for 6 months. After 180 days of storage, maximum retention of alpha tocopherol was observed in inbred line HKI323PVE. Maximum mean lipooxygenase activity was observed at 120 days after storage. The expression analysis of LOX3 gene showed similar trend as of absolute lipooxygenase activity throughout the storage period. Therefore, LOX3 gene can be used as a promising candidate for enhancing post-harvest storage. MSc student Mr. Shashidhar worked on genetic variability and molecular characterization for resistant starch in maize. Based on evaluation at 3 locations, he identified some of the inbred lines which are having 37 to 39% of resistant starch. Based on molecular characterization of inbreds through SNP markers, the inbreds were classified into 6 groups. Through 7x7 half-die allele analysis, it was observed that additive variance was predominant for amylose content and non-additive gene action for resistant starch. Mr. Brajesh Mehta, PhD student, worked on enrichment of seed corn genotypes with provitamin A, lysine and tryptophan through marker-state breeding. The improved genotype showed 5-fold increase in provitamin A, while 1.7 and 2-fold increase was observed in lysine and tryptophan respectively. The expression analysis of CRT-RP1 gene showed maximum expression in improved line at 24 days after pollination. Similar trend was observed in accumulation of provitamin A. The expression of OPEC2 was downregulated in improved genotypes and thus enhanced the accumulation of lysine content. Through biochemical analysis of harvested product, it was observed that highest Brix value was at 24 days after pollination, while highest lysine, tryptophan and provitamin A content at 20 days after pollination. PhD student Mr. Ratan worked on identification of QTLs and candidate genes associated with enhanced grain, zinc and iron concentration in wheat. He used biparental population as well as GWAS panel for mapping. He used SNP genotyping and ICA mapping for the analysis. In biparental population, he identified 3 major QTLs with phenotypic variance up to 8.1% for grain zinc content. In addition, he identified 3 major QTLs for grain iron content having a phenotypic variance up to 10.2%. Some of the putative candidate genes identified in QTL interval are ABC transporter, protein kinase, G protein, amino acid transporter and peptidase superfamily. Similarly, in GWAS panel, he identified QTLs for grain iron content having phenotypic variance up to 12.62%, while for grain zinc concentration, QTLs having PVE up to 7.6% were identified. MSc student Mr. Revant worked on genetic and molecular analysis of cooking parameters in rice variety Boka Chawal, also known as zero fuel rice. Factors affecting cooking time in rice varieties Boka Chawal, PB1121 and PB1509 were studied. Significant variation was observed for cooking time of milled rice among 3 varieties. In F2 derived F3 population, variation in cooking time showed a normal distribution indicating quantitative nature of the trait. Through BSA, linked markers could not be identified, therefore further studies are required. 6 MSc and 3 PhD students worked on genetics of miscellaneous traits. MSc student Mr. P.K. Nayak worked on morphological and molecular characterization of mutant lines in wheat. He used 250 selected mutants of wheat varieties SD3086 and SD3118, and mutants showed significant variation for various agromorphological traits. He identified some of the mutants for early heading, onless, and resistance to yellow and leaf rust. Mutants were characterized through SNP genotyping. The non-glaucous lines revealed deletion in DMC locus. Similarly, they identified a deletion of 25 base pair in onless lines. MSc student Ms. Baby worked on development of non-necrotic backcross derived lines in wheat. Published molecular markers for NE1 and NE2 genes were validated on positive and negative lines. Marker state backcross breeding was used to transfer recessive NE1 allele in wheat varieties SD2967 and SD2733 from the variety SD2932. Recovery of recurrent parent genome was up to 95% in BC2F1 generation. PhD student Ms. Archana worked on mapping QTLs for stay green and its component traits associated with nitrogen use efficiency in rice. She used a real population derived from the cross USA 677 and PSG16. The lines were evaluated in alpha lattice design. The major findings of the study are: stay green trait in PSG16 showed a monogenic inheritance pattern. Its major component trait, chlorophyll content, showed dominant and additive variations. A major QTL, QSG 1.1 for chlorophyll content was mapped on chromosome 1, which was associated with % flowering time. Significant associations were found for yield, stay green and NUE related traits. Mr. Jayant K, MSc student, worked on allelic distribution of white compatibility gene in rice. A total of 967 genotypes, which includes 180 tropical japonica lines, 297 international rice germ class lines,
and 490 germplasm lines were used to screen for S5 locus using Intel S5 functional marker. Out of 967 genotypes, 171 genotypes possesses neutral allele, 656 were indica type, and 140 were japonica type allele. Based on molecular characterization of 71 SSR markers, total germplasms are grouped into two clusters. Evaluation of hybrids between 41 white compatible varieties with 2 indica and 2 japonica lines showed no significant differences between indica and white compatible varieties and japonica and white compatible variety hybrids. Ms. Sahana Patil worked on inheritance and validation of markers for kernel row number in tropical maize. For genetic analysis, F2 population was used between parents AI 505 with 12 kernel row number and AI 542 with 22 kernel row number. Major findings of the study are: kernel row number exhibits oligogenic behavior, kernel row number has high heritability, kernel row number has a positive and significant correlation with ear girth and grain yield, and marker BNLG 1017 was found as linked with a kernel row number. PhD student Mr. Nitish Prakash worked on genetic analysis and identification of loci governing prolificacy in Sikkim primitive and novel landrace of maize in India. For genetic analysis of prolificacy trait, two F2 derived F3 populations were developed and evaluated at 3 different locations. The results indicated a major locus responsible for prolificacy in Sikkim primitive. Mapping of prolificacy trait in both the populations identified a major QTL having R-square value of nearly 30%. In the QTL interval, some of the candidate genes like grassy tiller 1 have been identified which needs further validation. Ms. Tejaswani, MSc student, worked on genetics and interrelationships of time of flowering and stem growth habits in chickpea. Genetic analysis in F2 and F2 derived F3 families showed that the time of flowering in early flowering and podding genotypes BG1044 and BG1054 is controlled by a single gene. Genes for early flowering in BG1044 and BG1054 were found to be allelic. Genetic analysis for semi-determinate growth in early flowering of poddy genotypes BG1044 and BG1054 showed a single gene for semi-determinate growth. Genes for SDT growth in BG1044 and BG1054 were found to be allelic. MSc student Mr. Raju worked on genetic analysis of plant height in Kajanas Kajan. Genetic analysis in F2 population revealed a single recessive gene for dwarfness. SNP genotyping of parents and contrasting bulls identified a putative candidate gene ABC transporter which may be responsible for dwarfing in ICP 14459. PhD student Ms. Manisha Saini worked on genetics and molecular mapping of seed viability in soybean. Genetic analysis revealed that more than one gene is involved in controlling seed viability in soybean. Two major QTLs were identified for seed viability with phenotypic variance of almost 11%. Markers SAT538, SAT316 and SAT173 could effectively separate the high from the low viability genotypes. Some of our faculty members and students got recognitions and awards. Dr. Rajbir Yadav was elected fellow of NAS, while Dr. P.K. Bhaumik and Dr. M. Vignesh became NAS associates. Our students Ms. Ashwini and Venkat Prakash Reddy were awarded IRE merit medals, while Ms. Bhuvneshwari was awarded with Assam Government Medal. During the year, 3 faculty members were added to the discipline of genetics and plant breeding. And as far as publications are concerned, 22 research papers were published from the research work of PhD and MSc students, and all these papers are published in journals with NAS rating of more than 6. Out of these 22 papers, 7 papers are published in journals with NAS rating of more than 10. As far as placement of our students is concerned, 7 MSc students, they are doing their PhD, and 8 PhD students got selected as scientists in ARS, BAR or ICFRE. With this, I end my presentation. Thanks.
Yeah, thank you very much, Professor Binod, for your excellent presentation, as well as completion within stipulated time. I am really sorry for the initial some disturbances. Chairman, sir, may we have comments at the end after completion of all these 3 presentations?
Yes. I think let others complete and we can have finally.
Okay, sir. Thank you, sir. So next presentation we will have from plant
Please go
genetic.
ahead. Please go ahead.
Yes, sir. Next presentation we will have from plant genetic resources. Professor Bina Gupta will make presentation.
Good morning to all, respected Chairman, Dr. Kuldeep Singh, Dr. S.P. Datta, convener, my dear friends, professors, students. I will be narrating you about the significant achievements made by the Plant Genetic Resources Division during this year. This year, we have 5 MSc students and 3 PhD students who submitted their thesis. The first student, Sandeep Kumar Panigrahi, worked with Dr. Kavita Gupta on the assessment of genetic diversity and differential reaction of black gram germplasm to pulse beetle Colesobruchus muculatus. He selected 69 diverse accessions, including 10 CWRs, that is crop wild relatives, along with 4 checks, and have screened the germplasm. And he has found 1 immune line, 1 resistant line, and along with the checks. The correlation heat map studies have shown that the growth index has significant positive correlation with adult emergence and seed weight loss, whereas seed hardness shows negative correlation with the adult emergence. The salient findings of his results are that IC 259504 of Vigna vexillata and IC 424616 of Vigna mungo were immune and resistance, which can be used in the breeding programs in further to develop bruchid resistance cultivars in plagram as well as the other Vigna related species. Second student Nagraj Nayak, he worked with Dr. K.K. Gangopadhyay on the genetic diversity analysis of wild brinjal germplasm of primary gene pool, Solanum insanum and Solanum incanum, and also their evaluation for the brinjal fruit and shoot borer resistance. So he screened the germplasm along with the checks, and this is the frequency distribution of important qualitative traits he found. Watts analysis have grouped all the germplasm into 5 clusters and 1 separate cluster for the check. Among the 5 clusters for accessions, 2 were exclusively for the Solanum incanum, which signifies that the clear-cut separation of the wild species from incanum, while the rest of the 3 clusters, they have both the wild species, but they were grouped as per their data. The significant findings of his work is the 7 accessions he have found of Solanum incanum and 1 from Solanum insanum, which were superior to the best check. And the molecular markers can detect a lot of variability within the incanum and insanum species, but no distinct separation of wild species of brinjal and cultivated varieties was seen. And these 3 lines have shown 0 fruit infestation. Third student Shubham Dev worked with Dr. Raj Kumar on cytogenetic and molecular studies to reveal the taxonomic status of Trichoxanthus cucumerina complex. He selected 35 accessions have grown in the greenhouse and studied the morphological variations in the seed as well as in the plant characters. This entry T11, it shows a different type of flower, and the salient findings are that the flow cytometry and the cytological studies revealed the diploid nature of all the accessions. So there was no significant variation. 10 out of 13 accessions showed 100% similarity with Trichoxanthus cucumerina variety cucumerina. T11 entry, which I showed you earlier, and T25, they form separate groups, means that they do not belong to Cucumerina var cucumerina. They may be related to the Trichoxanthus nervifolia. And T37 accession was not under this section. Maybe it is a natural hybrid between the two different species and may belong to a different section, that is Trichoxanthus brevibracteata. Fourth student, Mr. Mallikarjun Biradar, he worked with Dr. Sherry Jacob for marker-assisted assessment of genetic redundancy in selected maize germplasm collections. He selected 24 maize populations, Mimban landrace, which were from Mizoram area, 4 north districts, and he has studied the variability, like variability in tassel, variability in silk, green, pink, and red, then anthocyanin pigmentation of the root, leaf pubescence, plant height, and number of copse varying from single copse per plant to 3 copse per plant. Then intra-cluster analysis based on all these characters, he had shown that there was some redundancy in the accessions, and the salient findings of his work is that from 24 accessions, redundant set of 2 to 4 accession, total 12 were marked for bulking through sibbing. That means from 24, it reduced to 17, and then the variation in kernel color was not a significant character for explorers to collect the germplasm on this basis. And then within a district, samples collected from comparable altitude having synchronized flowering behavior and copse size, they should be pooled together. The fifth student, Mr. Praveen, worked with Dr. Sandhya Gupta on cryopreservation of Fragaria vasca, shoot tips, and virus elimination through cryotherapy techniques. So he selected this accession, which was conserved in the in vitro gene bank using V cryoplate technique and DAS, ELISA, and RT-PCR. He has standardized the cryopreservation protocol for Fragaria vasca, and these are some of the processes in the standardization. The salient findings are that out of 30 accessions, samples of Fragaria, which he tested for 4 viruses, 100% samples were free for these 3 viruses, whereas 90% were free for RP-RSV virus based on the DAS ELISA. And then post cryotherapy treatment, it had given very good results, 96.6% success for the virus elimination. Our PhD student, Mr. Anil Patidar, worked with Dr. Mahesh Yadav for unraveling the physiological and molecular mechanism of terminal heat stress tolerant in bread wheat, and he selected 96 bread wheat accessions, 79 indigenous and 17 exotic collections, 2 sowing dates, that is the non-stress environment in first week of December and heat stress environment in first week of January. And he has seen a lot of variability in the field, variability in ground cover, variability in plant waxiness, and some of the accessions remain green even at the maturity. The reduction in the grain size was also significantly, it was higher in the susceptible accession than in the tolerant accession. Also, the heat stress caused damage or it reduced the size of the starch grains and the cells in the aileron layer and the endosperm. And the salient findings are that the ultrastructural studies, they confirmed that endosperm and aileron layer of mature grains using SEM studies, that it was reduction in the size and density of starch grains, and it resulted in the loss of yield and the total grain weight in the heat stress susceptible lines. Then 24 primers pair amplified 263 alleles with 91.1% polymorphism, and then in total 35 unique alleles and 47 rare alleles were identified, and these can be used for DNA fingerprinting and genotype differentiation. The most stable and high yielding accessions were identified as climate smart germplasm lines. Mr. Jagdish Goenka, he also worked with Dr. Mahesh Yadav with the genetic characterization and allele mining of quality traits in maize landraces from Himalayan region. So he has selected 99 maize landraces, mainly from the 62 districts of 10 states of India's northeastern and northwestern Himalayan region. The phenotypic variability in ear size and shape was observed, and it was seen that the collections from northeast region, they have more average values for most of the quantitative traits, whereas the accessions from northwest Himalayan region, they have higher mean values for traits like leaf length, ear length, width, and kernel per row. Beta-carotene alleles, the favorable alleles, have also been identified in few accessions, and the major findings are that maize accessions of northeast region possess higher morphological diversity as compared to the accession from northwestern Himalayas. And this CRT RB1 favorable allele 2 600 pair base pair was identified in 4 land races, and these are 2 from the Himachal Pradesh and 2 from the Nagaland area. Mr. Hari Prasad, he worked with Dr. Veena Gupta for the agromorphological, nutritional and molecular diversity analysis of grain type of chenopods. So he selected the Chenopodium germplasm along with the indigenous germplasm along with some of the accessions from the exotic collections. The different species, it included 5 species. And this is the type of variability he has observed in the stem branching, the stem anthocyanin content, leaf shape, leaf color, and anthocyanin content within the species as well as across the species, panicle shape, color diversity within the species in Chenopodium cunea and Chenopodium album. Biochemical nutritional studies, the correlation studies have revealed that moisture, starch, and carbohydrates are positively correlated with each other but are negatively correlated to the protein, fat, and ash content. So the salient findings are that all the parameters showed high range of variability among the sections of the same species as well as from the different species. And this correlation studies revealed that moisture, starch, and carbohydrates are positively correlated to each other. The Lynch pairwise distance have shown that closely related, these 2 accessions are closely related and they are from the same species, album, and from the same area, and the highest highly diverse accessions are these 2 with maximum distance. 6 of the indigenous and exotic accessions each were identified as promising lines for biochemical parameters. Then during the year we had also organized one declamation contest for the students commemorating the 75 years of India's independence. And here the 7 of the students participated, and Latif Bashir, Siddhant Ranjan Padi, and Nitika Kalia, they were declared as first, second, and third position. At present, we have 39 students total, 29 PhD and 10 MSc, and total faculty strength is 46. Thank you very much.
Yeah, thank you very much, madam, for your good and lucid, very good and lucid presentation. So we will move to the next presentation. Next presentation will be from Seed Science and Technology. Professor Monica Joshi will make presentation.
Respected dignitaries, colleagues, and dear students, a very good morning to all of you. Myself, Dr. Monica A. Joshi, working as a professor in the discipline of Seed Science and Technology, will now present the significant educational achievements in the discipline. The mandate of the discipline ranges from seed production technologies to seed testing, HRD, and availability through farmers' participation. We are a total of 24 faculty members well represented throughout the country, and out of 24, 14 are eligible for MSc guidance and 11 for PhD guidance. With respect to students, 17 MSc students and 31 PhD students are on roll. In addition, 7 MSc and 3 PhD students will be getting their degrees in this convocation. This is a list of the MSc students due for their degree receiving: Vikram Patil, Payal Mathur, Sushma, who also competed for gold medal from the division, Sumitra, Alana, Bhanu Verma, and Rashmita. With respect to PhD, the 3 students are Venkatesh, Parveen Patil, and Sunil Jadhav. Presenting before you the educational achievements theme-wise, the first theme on seed production technology, wherein Malana worked on the standardization of seed production technology of sponge gourd hybrid Pusa Shresht by using the parental lines DSG31 and DSG43 with the main objective to study the plant growth, flowering behavior, seed yield, and seed quality, and to evaluate the effect of plant growth regulators and to optimize the planting ratio. This is a brief view of the treatments and other observations recorded by Malana, and he reported the superiority of DSG with respect to vegetative parameters and DSG43 with respect to seed quality. 6 AM to 12 PM was better for hybrid seed production, and 3:1 ratio was found to be better for higher number of fruits and seed yield, whereas 4:1 ratio better for seed quality. Ms. Monica worked on the effect of planting geometry and micronutrient application on seed yield and quality of Parthenocarpic gynoecious cucumber variety Pusa Parthenocarpic cucumber 6 by using various experimental materials, evaluating in different seasons, different plant spacing, and different vegetative stage sprays, reproductive, and a combination of both. This is a list of the treatments and the observations recorded by Monica, and she found that planting geometry of 100 centimeter into 45 centimeter along with foliar spray with nano zinc plus boron at the rate of 1 milliliter per liter was effective for enhancing seed yield and quality. With respect to the second theme area on the seed quality assurance, Mr. Vikram worked on the effect of salinity stress on seed yield and seed quality parameters in soybean, with the main objective to study the effect on seed quality as well as on plant morphological, seed yield, and contributing traits by using 150 germplasm accessions and 6 released varieties. He also attempted to standardize the protocol by using 2 different methods, Petri plate and between paper method. And by evaluating under the Petri plate method, he selected the genotypes for pot culture based on correlation studies between the salinity tolerance index and the seed quality parameters. This table depicts the categorization of the sessions into various categories, and he reported that black-seeded genotypes were observed to have better salinity tolerance as compared to the yellow-seeded genotypes. With respect to the third theme area on seed quality enhancement, Mr. Bhanu Verma worked on the effect of nanoparticles on seed quality and storability in paddy, with the main objective to evaluate the effect of nanoparticles on seed quality parameters as well as on seed storability. He used 62 treatment combinations, well represented by 3 metal oxides, 2 formulations, and 2 controls. And he reported that with respect to seed quality, dry bulk zinc oxide in various concentrations was found to be better. Similarly, while studying the storability parameters, he reported that dry bulk zinc oxide, again at separate concentrations though, was found to be better when stored in polythene bag. However, when storability studies were carried up to even after 6 months, same polythene bag, when the seed was stored with theorem at the rate of 2 gram per kg, was found to be the best. Similar studies were done by Ms. Sushma on the effect of nanoparticles on seed quality, yield attributes, and storability in chickpea, with the main objective to study the effect on seed quality, seed yield, and the storage potential. The experimental material comprised of variety Pusa 547, chickpea variety, again in 62 treatment combinations. This is a list of the observations recorded, and she found that under laboratory conditions, dry nano zinc oxide at the rate of 250 ppm was found to be best. This was further validated by the field studies carried out by Ms. Sushma, and again it was found that dry nano zinc oxide at the rate of 250 ppm was found to be best. Even when storage conditions were conducted, after 6 months of storage, higher seed quality parameters were reported for the same metal oxide at the same concentration, and polythene bag was found to be better. With respect to the fourth theme area on seed biology, Ms. Payal Mathur worked on the studies on the basis of phenol color reaction in seeds of wheat, with the main objective to assess inter and intravarietal phenol color reaction and to understand the occurrence and to estimate the polyphenol oxidase using 25 wheat varieties. So these are the different wild species which were studied by her, and this depicts the estimation of different polyphenol oxidase using standardized methods, and she reported that grains in the middle portion of earhead showed higher intensity of phenol color reaction. In wild species, no effect of storage was found, and generally wild species showed higher PPO activity than that in the cultivated varieties. Second, with respect to Ms. Rashmita, she studied the studies on the seed quality parameters in relation to biochemical composition in Indian quality mustard, with the main objective to evaluate the seed quality, the biochemical parameters in freshly harvested and controlled deteriorated seeds, and to further study the correlation between them by using 30 genotypes of Indian mustard, including conventional, single zero, and double zero genotypes. So she reported that higher germination percentage, seedling vigor indices were recorded for conventional genotypes, followed by single zero and double zero genotypes. This is the view of the field emergence which was recorded by Ms. Rashmita, which validated her findings. And ultimately, she concluded that seed composition indeed affects the seed quality. With respect to the last theme area on seed pathology, undertaken by 3 PhD students of the division, first being Mr. Venkatesh MG, working on the impact of sheath rot of paddy caused by Cerocladium oryzae on seed quality, its diversity, and development of management strategy, with the main objective to study its role on seed quality, diversity studies, and the protocol for management strategy as told earlier. So Venkatesh collected the sheath rot samples from various locations across the country, and he found that the mean germination percentage for maximum samples with respect to healthy panicles was 77% as compared to the infected sample where the germination percentage was very low. He also used 19 gene targeted markers, and this represents the variability by few of them. Using a combined dendrogram, he found that the isolates could be grouped into 7 clusters, which were then represented by the various agroclimatic regions. He also did in vitro evaluation of fungicides against Cerocladium oryzae and found that carboxin plus therum was the most effective fungicide since it registered higher % inhibition. For the development of the management strategy, he reported that a combination of treatments and biocontrol agents was found to be best. With respect to the second PhD by Mr. Parveen Patel on the development of eco-friendly management strategies against grain discoloration for improved seed quality in paddy, with the main objectives for morphological and molecular characterization and to study the effect of seed discoloration on the seed quality parameters and to develop eco-friendly management practices. He identified a total of 9 fungi which were associated with seed discoloration and found that Alternaria alternata and Fusarium equisiti were present in majority of locations. This depicts the difference in the healthy and discolored seed. This is the difference between healthy and discolored under a stereo microscope. With respect to molecular characterization, the confirmation of the pathogen was done by doing multiple gene analysis, and totally 6 groups were formed based on the phylogeny. For the eco-friendly management practices, he reported Trichoderma viridi and Trichoderma hertzianum performed best among biocontrol agent, and under field condition, Trichoderma viridi performed best with seed treatment, seedling dip, and spray. With respect to the third PhD by Mr. Sunil Jadhav on the study of Fusarium oxysporum on seed quality detection and development of wilt management strategy for quality seed production in lentil, with the main objective for characterization, development of specific marker, and management strategy. He surveyed and collected lentil wilt infected seed samples from various locations across the country. This is a representation of the cultures, difference in the healthy and the infected seed with respect to seed germination. And he also reported that out of 25 primers which he screened, 6 primers showed reproducible polymorphism and the isolates were grouped into 4 major clusters. Similarly, under field conditions, seed treatment with carbendazim and mancozeb were found to be best, and he reported that chemical treatment using combi product of fungicides was the most effective. With respect to the publications from our discipline, we have 36 research publications, 4 training manuals, 1 practical manual, 2 souvenirs, and 27 and 5 manual and book chapters respectively. This is a list of the student publications. I have listed with NAS rating more than 6. With respect to student placement, our students have been placed in very well organizations by the dint of their hard work, and 5 of our students have enrolled for the IRI PhD program in the institute itself. This is a list of the courses being undertaken by our faculty, very dedicated faculty, and then we also are regularly involved in paper setting, thesis evaluation, viva voce, and oral exams. This is a glimpse of the 4 online trainings organized by our division. 1 training organized at IRI Regional Station Wellington by our outstation faculty, Dr. P. Nalathambi. This is a glimpse of the extension activities taken by our faculty in the IRI itself, along with demonstration of the IRI technologies and varieties at IRI Regional Station at Pusa, Bihar by Dr. Aki Singh. This is a list of the PG activities, development of a practical manual, participation of our students in extracurricular activities. With respect to the equipment procured, the funds which are allotted for PG are generally diverted towards the development of the divisional classroom and the PG laboratory. This is a glimpse of the honors and awards by our faculty in various conferences. Dr. Atul Kumar, myself, Dr. Sangeeta Yadav, Dr. Shiv Kumar Yadav, Dr. Lal, Dr. Atul, Dr. Nagmani, and Dr. Nala Thambi. This is a list of the awards won by our students in various seminars and conferences. And lastly, the recognition with respect to various panelists and members, invited lectures, and few other as national level recommendations, designated members, and other institutional responsibilities. This brings me to the end of my presentation, and I would like to acknowledge our Director Sir, Joint Director Research, Dean Ma'am, and Joint Director Extension, along with all ICR and IRA authorities. And I would also like to place on record my gratitude to the students, faculty, and staff of my discipline for all their needed support. And I would like to end by this remark that education is the passport to the future, for tomorrow belongs to those who prepare for it today. Thanks one and all.
Thank you very much, Professor Monica. So, Chairman Sir, with this, we could complete all three presentations. Now, how to go about, Director Sir? Should I invite comment, or due to paucity of time, I invite the remarks from Chairman?
It is Chairman's
Yes,
remark.
Chairman's.
Okay, okay. So now I invite the remarks from Honorable Chairman, Dr. Kuldeep Singh Ji. Sir.
Thank you, Dr. Respectable Dr. Aki Singh, Director IRI, Dr. Rashmi Agrawal, Dean Postgraduate Studies, Dr. Manjaya, Dr. Datta, Dr. Chakravarti, Dr. Vishwanathan, Dr. Veena Gupta, Dr. Monica Joshi, and I see at some time 480 participants. Now this is 671 now. So that shows the interest of the students in this one. First of all, congratulations to the students and the faculty of IERI for the 60th convocation. And while I was at NBPGR, I enjoyed this week for all the 5 years. I think this was the best time to learn from students in the shortest possible time. And this happened today as well. I could really have a glimpses of 39 different theses in crop improvement division. Congratulations to all the 39 students of crop improvement who have completed their master's or PhD thesis and are eligible for the award of the degrees and probably entering a new phase of their life. Dr. Aki Singh, I have seen, and of course, this is not a secret at all, that IRI is the best temple of learning throughout the country and probably one among the best in the world. There's no second thought on that one. The sort of research work I have seen, different, so diverse in terms of crops, in terms of the areas of research. And I am sure most of the PhD thesis the students might have taken as per their own interest. It's not only the basic research which yielded into a number of publications. I think I could see a very large number of publications coming out of this thesis and very high standard publications. But I also see some of the lines entering directly into the practical use. USA 44 lines with different QTLs for drought, and for monogenic, digenic, tetragenic lines, allelic lines. They are not material only for evaluation under ICRIP, but this will be a basic material for several other studies. So I will not comment more on those one. I know there's a paucity of the time. It's a wonderful seeing the good work of all the students, but I will make one suggestion here, Dr. Singh. We have the best students and the best faculty at IERI. And all of us have seen with the COVID-19 pandemic that we have been depending for our food system on only a few crops. And in fact, might be relying only a few crops might have changed our overall nutritional status. You are well aware that we as a nation are very rich in diversity, and there are large number of crops, what we call as potential crops. And though we have a network, ICRIP network to work on those, but that's dealing with the classical or the traditional breeding procedures. All those crops have a good potential, but they have a lot of limitations, and not much of the things have been worked out. So I will request you, Dr. Aki Singh, that being director of the IERI, please think over taking up a couple of potential crops in IERI fold so that you can put some of the best students on those crops and really help the nation in bringing about a change in the nutritional status of the population as a whole. And good number of these crops are not only rich in nutrition, but they are really climate resilient because they are growing in harshest of the environments. I'm not talking of the crops with the ICRISAT is working. They are all also different and very different nutritional. They are the dryland crops. But in addition to these crops, we have a good number of crops. For example, NVPGR is working, the ICRIP, we have the potential crops on 16 different crops. And probably you can have an internal thinking, internal review, maybe invite NVPGR to ask them how many crops those are. And if 4 or 5 are picked as the crops where this basic studies where we are lacking, those are also taken up. Wonderful work by all the students. Congratulations to all the students who will be really awarded degrees, 39 students in the crop improvement, and there may be more than 100 students in other disciplines, to all of them. With this one, I thank you, Dr. Yadav, Dr. Aki Singh, for giving me this opportunity. And in 1 hour, I learned more than probably I would have learned in several years together. Thank you. Thank you very much.
Thank you, Dr. Thank you so much.
Yeah.
Thank you so much.
Sir.
We will take care of your suggestions. Thank you so much.
Sir, we have seen Dr. Jalali has joined, sir. So
Yeah.
on behalf of Director IRI and on my own behalf, I would like to welcome Dr. B.L. Jalali.
Welcome, sir.
Next, welcome,
Very
sir.
good morning and welcome to you.
And Dr. H.S. Gord, also former Dean IRI. I
Yeah.
see him also, sir. Welcome, Dr. Gord.
Okay.
We will proceed further.
Okay. Yeah. Thank you very much, Chairman sir. Before moving to the next session, on my own behalf and on behalf of the institute, I am highly grateful to Dr. Kuldeep Singh ji, former Director NBPGR, Cluster Leader Gene Bank and Head Consultant ICRISAT, New Delhi, for kindly agreeing to our request to grace this session as a chairperson. Your vast experience in the area of plant breeding, biotechnology, as well as plant genetic resources greatly benefited our faculty as well as students. And I am sure your suggestion and critical remarks will help in shaping our future research. Thank you very much, sir. And I also thank all 3 speakers, Professor Binod, Professor Bina Gupta, Professor Monica Joshi, for excellent presentation, that too within stipulated time. Thank you. Thank you very much. So, Madam, please.
Yes, thank you very much. And I would also like to thank the chairman of the previous first session, Dr. Kuldeep Singh, and all the speakers, professors who made a very meticulous and very nice presentation, very crisp, that they finished everything well in time, rather 2 minutes before time. So thank you, each one of you, and keep joining for the next session. And again, good morning to all of you. And it's now we are entering into the second session of the third day of 60th Convocation Week program. And I would like to again say that now again we will be looking forward the presentations made under the School of Crop Protection. And these will be the significant educational achievements made during the year by the students of MSc and PhD students of 4 disciplines, which are under the School of Crop Protection. And these disciplines are, we will have series, we will have first the agriculture chemicals, entomology, nematology, and plant pathology. So on this second session, I warmly welcome Dr. B.L. Jalali, former Director Research, Haryana Agriculture University, Hisar. And all of us know that Dr. Jalali had been a very guiding force, and with all his novel ideas, he had been guiding all the 4 disciplines of School of Crop Protection. And you all recall that he was also the part of the accreditation team who visited our institute last year, and we got re-accreditation under his guidance and under his support. So thank you so much, sir, for accepting the invitation and coming here and guiding us, and keep guiding us for many more years. So at the same time, I would like to welcome our director, Dr. Aki Singh, who all of you know that he is guiding, and he is a pillar. And all these programs without his guidance, we wouldn't have been reaching to this stage where we are now. And at the same time, I would like to welcome Joint Director Extension, PD, Dr. Man Singh, all heads of the division, professors, faculty, students, and ladies and gentlemen. So this School of Crop Protection, total there are 48 theses. And let me tell that the students' significant contributions are not the total part of research achievements being done or being carried out at these 4 disciplines. It's only part of the research work which is being carried out. So with these introductory remarks, now I would like to have initial remarks of our director, and then subsequently floor will be Dr. S.P. Datta's.
Thank you, Dr. Asmi, and I would also like to add my own words of welcome to Professor Jalali. And to all of you, Professor Jalali is very well known in academia for being very critical in his approach. He is a proud alumnus of Banaras Hindu University, having done both bachelor and master's degree, and PhD from Punjab Agriculture University. And he served as professor and head and researcher in the Department of Plant Pathology at Haryana Agriculture University. Also, for a brief period, he was director of Center for Integrated Pest Management and director of research at Haryana Agriculture University. He served also as visiting professor at a number of universities: University of Hanover, Tübingen, University of Montreal, Bangkok in South Korea. And his research interest is primarily has been in understanding the dynamics of mycorrhizal association under biotic and abiotic stresses, decision support system in integrated pest management, and molecular biology of host pathogen interaction. One of the projects on which he was guiding us, supported by NAIP, where we had a mega project involving multiple centers on host pathogen interaction, particularly in relation to blast, of which Dr. T.R. Sarma was PI. And that period of 5-6 years was a great learning period for me in particular, the way Dr. Jalali used to guide us for taking the research objective of the project to a logical conclusion. He has received a number of awards, including Narasimhan Award and Paugi Award. Professor Paugi, MS Paugi, is a well-known figure in plant pathology, and also Sir J.C. Luthra Award. He has been a very important resource person in several committees, whether it is ICR, DVT, DST, and RDC, UGC, and providing his guidance in project evaluation, project monitoring, mentoring. So that has been a real great contribution from Professor Jalali. He's fellow of National Academy of Sciences and Indian Phytopathological Society and Indian Society of Plant Pathology. We have great pleasure, sir, in welcoming you for this session, which is going to be a compiled presentation of the students' research work. I must say that yesterday, students from each discipline competed for the best student medal and the IRI medal. And then the remaining students who have completed their master's and PhD degree in these disciplines, their work is no less significant. When there is an award, only it will go to only one, but doesn't mean that the others have done any less quantum and quality of work. They are all equally good. So their work, there is a compiled presentation going to be done by the professors of respective discipline. And based on those presentations, we would like to have, finally your comment and guidance as to how our research program should move in future, considering the coming up challenges. So once again, I welcome you heartily, sir. Thank you for sparing your time and for being always available. Whenever we request you, you are there. Thank you so much.
Thank you, sir, for your kind remarks and initial remarks. So we will move to the second school, that is a School of Crop Protection. As we know, this is also very important school, as annually about 30 to 35% of our agriculture produce gets wasted because of the various pests. And it comprises of 4 disciplines: agriculture chemicals, entomology, nematology, and plant pathology. So now, with the kind permission of Chairman Sir, may we take up the first presentation?
Please go ahead. Please go ahead.
Okay, sir. Thank you very much. So first presentation will be from agriculture chemicals. Professor Neera Singh will make presentation. Thank you.
A very good morning to all. Respected Chairman of the session, Director, Joint Directors, students, ladies and gentlemen, I am presenting significant educational achievements of postgraduate students of discipline of agricultural chemicals. Division has mandate of development of agrochemicals, their formulation and safety evaluation, as well as human resource development. Division has 15 faculty members and 41 students on roll. Ashish Khandelwal, PhD student, and Anirban Sil, Ranjit Kumar, Shyam Kumar, Renu Saini, Biswajit Horizon, and Sumit Shekhar, MSc students, will be getting their degrees in 60th convocation. Theses have addressed the theme area of decontamination, development of agrochemicals or formulation, and analytical methods. Ashish Khandelwal addressed the issue of crude oil contamination of agricultural field using bioremediation technique. 17 bacteria and 3 fungi were isolated from a crude oil contaminated soil and isolated microbes were characterized. Among these microbes, a consortium of bacteria Pseudomonas and Bacillus amyloleucifens and fungus Aspergillus sedovi were identified as best crude oil degrader, while Cocoria rosea and Aspergillus sedovi consortium was identified as best pH degrader. These microbes were formulated in 3 different types of bioformulations. Free and bioformulated microbial cultures significantly enhanced crude oil or pH degradation in soil as compared to control soil. Supplementing soil with ammonium sulfate enhanced degradation. Guargum nanobentonite composite was identified as the best bioformulation for crude oil or pH degradation. Further, these microbes were highly effective in degrading crude oil in a naturally contaminated soil and decreased half-life by 2.3 to 2.8 times. Finally, this study concluded that these microbes can be used for crude oil and pH degradation in contaminated environment. Anirban Sil, a MSc student, prepared magnetite-based composite for pesticide removal from water. 18 composites were synthesized and were screened for the removal of 12 pesticides. Based on the removal efficiency, 4 composites were shortlisted and were characterized. Further, based on optimum sorption, regeneration, and reusability, 2 adsorbent MAG 15 and MAG 18 were identified as best adsorbents. Further, these 2 adsorbents showed 98 to 100% pesticide removal from contaminated tube well water and Yamuna river water. This study suggested that these 2 adsorbents have potential for their use in pesticide removal from contaminated water. Ranjit Kumar evaluated insecticidal properties of Xanthoxylum alatum seed extracts. Essential oil and hexane extract fraction from the seed were characterized. Both essential oil and hexane contain linalool and limonene as the major constituent. Nanoemulsions of essential oil and hexane extract were prepared and they were characterized. These nanoemulsions showed good acaricider and antifeedant activity. Thus, this study suggested that essential oil and hexane extract from Xanthoxylum alatum can be exploited for their insecticidal use. Biswajit prepared and evaluated control release product for pest control in 00 mustard. Essential oil from 4 mustard varieties and 14 commercially available essential oils were evaluated against Tribolium castanum and Corsaira cephalonica. Bioassay by fumigation method suggested that mustard, garlic and metal essential oils are potential bioactive oils. These essential oils were formulated in different combinations. A combination of 3 essential oil gave better result than binary combination. A sachet formulation of 3 essential oil showed 100% control of both pests at half dose. Thus, these studies suggested that mustard, garlic and metal essential oil formulation can be used for managing storage pest of mustard. Shyam Kumar assessed alkaloid based formulation for thrips management. In silico screening of 7 alkaloids against 4 target protein, identified that tropinone was the best alkaloid. This was further confirmed by in vitro screening against thrips palmi. Identified tropinone alkaloid was formulated in chitosan matrix and 9 formulations were prepared and evaluated against thrips palmi. Results suggested that CIP 500 formulation was best in terms of encapsulation efficiency, loading efficiency as well as the pest control efficiency. So finally this study concluded that alkaloid tropinone has potential for its use for thrips palmi control. Renu developed and evaluated azole-based nanofungicides for managing pathogens in maize. 6 fungicides were nano-sized and evaluated against 4 fungi. Results suggested that compared to pure AI, nano-sizing resulted in 30 to 50% reduction in ED50 values. Tabuconazole was identified as the most active compound. In vitro evaluation in net house condition further confirmed the effectiveness of these fungicides against Rhizoctonia solani. So this study suggested that nanofungicide formulations have potential for their use in disease management in maize. Sumit evaluated matrix effect on pesticide residue estimation in crop group 27 and 28, which is the herbs and the spices. An analytical method was developed and validated for 100 pesticides in the representative crops of 27 and 28 crop group and was evaluated in the member crops. Results suggested that methods developed in representative crops coriander, curry leaves and cardamom were very good in pesticide residue estimation in member crop, that is mint, tejpatta and tamarind. However, method developed in cumin was not good for pesticide residue estimation in fenugreek. Therefore, this study suggested that along with taxonomical similarity, matrix effect should be included as a parameter for crop grouping. 36 research papers including 13 student papers were published last year. Ashish, Randeep, Shrosi, Rajni and Anirban were honored for their academic and extracurricular activities. Finally, I would like to thank all students, their chairperson, faculty members, students of discipline of agricultural chemicals for their contribution in my presentation. Thank you everyone.
Thank you very much, madam. If chairman sir permit, we will go for next presentation, sir.
Sure, please go ahead.
Okay. Thank you, sir. Next presentation will be from entomology, and Professor Devjani, they will make presentation.
Good morning, everybody. With pleasure, I present the significant educational achievements of the Division of Entomology. The mandate of our division is to conduct basic and strategic research, to develop as a center of excellence, and to provide services in insect science and technology. 17 of our students will be receiving their MSc and PhD degrees this year, and all the areas of entomology have been addressed by them. Mr. Varun Seni has undertaken taxonomic studies on tribe Crabronini under my guidance. He has collected specimens for his study from various locations in India, and also the collections of various museums were also studied. 9 species were recorded from the first time from Delhi. During his studies, he also established a new report for India, that of Crossoceros quinlani. A checklist for the species of tribe Crabronini was compiled, and it comprised of 13 genera and 88 species from India with updated lists and new locality records. 12 new species were described from India, and 3 subgenera were recorded for the first time from India. A phylogenetic tree based on morphological data was also constructed, and it can be clearly seen from the tree that this tribe is a paraphyletic. Ms. Komal worked on the Macrolepidoptera of Pusa campus under the guidance of Dr. Shpiya. With the help of light traps, Ms. Komal collected specimens from 8 different locations in our campus, and also did host-based rearing of the Macrolepidopterans. A checklist of moths of Delhi was compiled, and 331 species belonging to 32 families were included in the checklist, and Noctroidea was the most species rich, followed by Pyralidia. More than 180 illustrations were included in the 37 species of micro moths could be collected from Pusa campus, of which 28 species are new records. Ms. Ipshita worked on Brassica gentiana genotypes for elucidation of different mechanisms of resistance to the mustard aphid under the guidance of Dr. She evaluated 30 Brassica gentiana genotypes, both under natural and artificial infestation, and based on the resistance indices which she calculated, she could identify 6 of the lines with promising characters which could be used in the breeding programs. Significant increase in starch, sugar, protein, phenol, antioxidant, FRAP, and tannins was seen, while there was a decrease in the glucosinolates and plant pigments. Activity of all the test defense enzymes and macronutrients increased in response to damage by Lipophis erythemi in all the plant parts of the test genotype. Ms. Roshni Gimire, under the guidance of Dr. M.K. Dillo, studied the biological and biochemical interactions between Lipophis erythemi and Brassica gentiana. Under artificial infestation also, similar results were obtained. Therefore, it was concluded that these varieties could be used in mustard. Further, she has observed that phenols, tannins, glucosinolates, and APX play an important role in plant defense against Lipophis erythemi, as they tend to reduce the fecundity and Mr. Ashish Raut, he studied the impact of climate change on brown plant hopper population with off-season survival and host plant resistance under the guidance of Dr. Subhash Chandra. No trace of BPH survival was found in Delhi during off-season. However, he recorded BPH surviving in South India during the same period. Through molecular studies, he showed that a high degree of homogeneity was there between the populations which he had collected from different areas, different rice growing areas, and exact route of immigration could not be deciphered using MTCO1. Weather parameters influenced BPH population, and he could conclude that influence of weather parameters on population dynamics of BPH was location specific. He also studied the influence of elevated carbon dioxide on BPH, and he found that BPH had higher multiplication rate and sucking rate, and also due to the higher number of nymphal and brachypterous females observed on the plants, resulted in a much higher BPH population on all the cultivars. Elevated carbon dioxide also affected the biochemical parameters of the plants, and overall elevated carbon dioxide compromised and jeopardized the mechanism of host plant resistance in the cultivars studied. Mr. Yogesh Lele, again under the guidance of Dr. Subhash Chandra, worked on climate change impact assessment and ecological engineering studies on BPH. Elevated carbon dioxide alone and together with elevated temperature enhanced the growth parameters of both BPH and rice plants. However, elevated ozone had a negative effect on all the growth parameters. Stressed conditions of elevated carbon dioxide, ozone, and temperature decreased the protein and sugar content in the rice plants. However, the level of protective enzymes, catalase and peroxidase increased further after. Planting of crop plants and flowering plants around the rice fields increased the abundance and activity of natural enemies, and this significantly reduced the BPH population, and also the population of other minor pests were also reduced. Mr. Prabhulinga, again under the guidance of Dr. Subhash Chandra, studied the host plant resistance against rice BPH Nilla parvata lugens with climate change. He evaluated several rice germplasms against BPH and identified 2 of the lines as new effective sources of BPH resistance, which could be used further. The effect of elevated carbon dioxide in rice plants amended with silicon was also studied. Silicon amendment at various doses resulted in significant lower protein content, and also the activity of the stress enzymes was significantly higher. Honeydew excretion was higher under elevated carbon dioxide, but it was lower in silicon treated plants, and application of silicon resulted in lowering of the transpirational rate and stomatal conductance. and therefore water use efficiency increased. Mr. Ramesh studied the dynamics of endosymbiont interaction in Babesia tabaci under the guidance of Dr. Subramanian. He recorded 5 endosymbionts from Babesia tabaci, and infection frequencies of Rickettsia and Wolbachia showed declining trends in year 2019. However, no significant differences were observed in the infection frequencies in populations collected from various parts of India and also from different. Significant differences were observed in biological parameters of all the four populations studied. The total developmental time was lowest for Asia 2-7 biotype and highest in Asia 2-5 biotype. Studies on endophyte endosymbiont interactions were also conducted, and Bacillus and Pseudomonas species were found as the most common bacterial determinants in genetic groups. GFP tagging and confocal microscopy studies on the endophyte endosymbiont interaction confirmed the horizontal transmission pathway of microbiota between the plants and insects, especially in the cotton Babesia tabaci. Ms. Chaitra, under the guidance of Dr. Vinay Kalia, investigated the role of gut microbes in Bacillus thuringiensis toxicity against pink bollworm, Pectinophora gossypiella. 8 different populations of Pectinophora gossypiella were used for the study. 37 bacterial isolates could be isolated from these populations, and higher bacterial diversity was recorded in the Adilabad and Khandwa populations, and males had diverse gut bacteria compared to the female, and Penochrobacterium glaciae was a new report from the insect gut of this insect. The metagenomic data was also obtained through outsourcing. The functions of the unculturable gut bacteria residing in the midgut of Pectinophora could be biofertilization and bioremediation. Antibiotic sensitivity assay was performed with the bacteria isolated from the gut, and 2 of the antibiotics used, tetracycline and chloramphenicol, showed 100% sensitivity, and they were further used for elucidation of the mechanism of variation in susceptibility, and a difference in mortality between the treatments of toxin alone and in combination with antibiotic mixture in bioassays was seen. And however, these studies were on the population from a single location. Further confirmation is required. So Neeta Pola again worked under the guidance of Dr. Vinay Kalia on the colonization of entomopathogenic bacteria in brinjal plant and interaction with Leucinodes orbonalis. Among the 25 bacterial isolates studied, 3 were identified with potential, and standardization of inoculation technique for colonization of the identified potential BT strains was done, and the cry gene confirmation in BT colonies recovered from leaf samples was also worked on. GFP tagging could detect the fluorescent bacteria in the stem and leaf tissues. And the leaves taken from the colonized brinjal plants were able to cause up to 44% mortality when they were fed to the neonates of leucinodes. So this is the first report on endophytic ability of native BT strains in brinjal and its effect on larval. Mr. Rudra Gowda, under the guidance of Dr. Subramaniam, characterized the gut bacteria of Italian honeybees Apis mellifera. The cultural aerobic gut bacteria associated were found to belong to 3 of the phyla: Firmicutes, Proteobacteria, and Actinobacteria. And Bacillus was the most dominant genus, and 7 novel gut bacterial genera also could be identified. The possible roles of gut bacteria could be hydrolysis of macromolecules and also their association with digestion and absorption. Deepak Mahanta worked under Dr. Amalendra Ghosh, and his title of his work was Identification of Thrips Pummy Genes Responsive to Tospovirus Infection. Comparative transcriptome of Thrips pummy in response to groundnut bud necrosis infection identified 2365 differential DEGs, and out of these 1384 DEGs were upregulated and 981 DEGs were downregulated, and majority of these were involved in innate immune response, cuticle development, receptor signaling, and psychotic pathways. The expression of 13 putative genes were in response to GBNV infection were validated further by RT-qPCR, and this study improves our understanding of networks of molecular interaction between Thrips-Pummy and GBNV and suggests potential targets for generic pest control. This could be published in a high-rated journal with impact factor of 11.64. Online data sets were generated. Ms. Isai Muddini, under the guidance of Dr. Sagar, investigated the impact of thermal stress on reproductive physiology of Spodoptera frugiperda. The adults of Spodoptera frugiperda were exposed to a single hot event for different durations, and this affected their fecundity negatively in male-only stressed combinations, irrespective of different durations, and female-only stressed combinations showed higher fecundity. Mating success and mating frequency of 4 combinations were also studied. The success percentage of mating decreased with increase in duration in only male stressed combinations. Expression of level of heat responsive and reproductive genes by qRT-PCR was also attempted. The expression of heat responsive genes decreased with increased duration in male adults, whereas in females the expression pattern of the same gene was inverse. The expression pattern of juvenile hormone esterase was similar in stressed male and female adults, showing upregulation at 2 and 6 hours and downregulation at 4. Ms. Arti, under the guidance of Dr. Amalendra Ghosh, studied the changes in transcriptome of whitefly Bemisia tabaci associated with begomovirus infection. Comparative transcriptome of Bemisia Asia 2-1 population in response to chili leaf curl virus infection showed upregulation of 29 DGs and downregulation of 51 DGs, and Go analysis indicated majority of the DGs to be involved in biological processes, and this work could be published with high NAS rating. And expression of 20 highly upregulated and downregulated putative genes of beta bacilli were accessed by RT-qPCR, and this study increases our understanding of the molecular interactions between the whitefly and the chili leaf curl virus infection, and suggests also potential genetic targets for the pest control. Data sets were uploaded, and data generated from this study will enrich genomic information of whitefly and will further aid functional studies. Ms. Sujata, under the guidance of Dr. Sagar, studied the physiological response of Spodoptera litura exposed to long and short-term thermal stress. Some of the salient findings were larval stage exposed to thermal stress at 46 degrees centigrade resulted in more number of female adults, and fecundity of adults emerging from stressed eggs decreased with increasing temperature and duration, and multiple thermal stress severely affected period of oviposition. Metabolic response of Spodoptera litura exposed to thermal stress revealed that adults emerging from stressed egg and larval stages showed increased protein, carbohydrate, and glycogen levels irrespective of the sex of the adults. And level of metabolites was more in female adults than the males. So it can be concluded that temperature stress reduces the survivability of the insects, and fitness of the survived individual is also affected. Sudhakar Kelagiri, under the guidance of Dr. G.K. Mahapatra, studied the toxicity variations of insecticides against cotton whitefly, and 7 whitefly populations from major cotton growing regions of India were collected and brought to the lab, and insecticides tested were fipronil, flunicamid, buprofezin, spiromesifen, and pyriproxyfen. And adults of Bemisia tabaci from Sri Ganganagar and Amravati population showed high to moderate resistance to both fipronil and flunicamid, and at nymphal stages all the populations were moderately resistant to both the above insecticides, whereas were susceptible to the other 3. The detoxifying enzymes were also quantified and expressed, and significantly higher levels of detoxifying enzymes were recorded in Sri Ganganagar and Ludhiana populations, and significant correlation was observed between LC50 values of all the insecticides and cytochrome and GST activities. Studies on sublethal doses of insecticides on feeding and reproductive potential of bee tabaci was also done, and it could be concluded that all sublethal doses significantly reduced honeydew production, and flunicamid was found most effective, and pyriproxyfen was most efficient. Ms. Sangeeta, under the guidance of Dr. Suresh Nebapure, conducted studies on efficacy of phosphine and contact insecticides against Rhizophilus surinamensis and Cadra cotella. Fumigant toxicity of phosphine was dose dependent, and with increase in exposure time, concentration of phosphine required for mortality decreased, and the order of toxicity in case of Rhizophilus surinamensis was maximum in the adults, followed by 15 to 20-day-old larvae and 5 to 10-day-old larvae. In case of Cadra cotella, the order of phosphine toxicity was egg more than pupa, more than larvae, more than adults. Contact bioassay for different time exposure periods was also done, and fenaprir and lambda cylohydrin were effective against adult and larval stages of Rhizophilus surinamensis, and deltamethrin and lambda cylohydrin were effective against adult and larval stages of Cadra cotella respectively. We could conclude from the present study that Indian population of Rhizophilus surinamensis might have developed some degree of resistance, and also contact insecticides other than deltamethrin and malathion, which were found effective, could be explored as alternatives. The faculty strength currently is 28. This is the breakup of the faculty. 17 research guides. 1 faculty was inducted last year. A number of students, after excluding the 17 outgoing students, is 71. These are some of the awards and honors won by the faculty. A workshop on microscopy was conducted for 3 days, and it was hands-on training for the students of our division. 13th S. Pradhan Memorial Lecture was also held. A 15 days workshop was conducted for the MSc students admitted during 2021, immediately after they came back to the campus, and they were given an exposure, and hands-on practicals were conducted on the important techniques for entomological research. These are the exposure visits of students which were conducted: 1 to IPFT Gurgaon, then visit to Farmer Ghaziabad, and visit to Institute of Food Security Gurgaon. The details of the fellowships held by our students: employment details of the students. The ARS scientists, 3 of them, they are all split PhD students. And publications through students' research this year: NAS rating more than 10.3, NAS rating more than 6.11, and NAS rating of 5 to 6 were also 11. These are some of the awards won by our students. And in addition to the students' awards, they also won in several extracurricular activities. I acknowledge the help received from Director, Joint Director Research, Joint Director Education, Joint Director Extension, PG School Administration, students and staff of the Division of Entomology. Thank you.
Hello. Hello.
Yes, Dr. Datta, you are audible.
Okay, madam. Thank you, madam. Thank you very much, madam, for your again excellent presentation, and that too within stipulated time. So now we will move to next presentation. Professor Matiar Rahman Khan will make presentation on behalf of Nematology.
I am presenting significant educational achievement of Division of Nematology during 2021. Our total student strength 32, MSc 9, PhD 23. In the 60th IRI convocation, 5 MSc and 3 PhD students are receiving degree. Our student research comes under the major themes nematode detection and diversity, nematode biosystematics, plant nematode interaction, silencing nematode genes, EPNs for insect pest management. Amit Ahuja, PhD student, worked on determination of genetic diversity of potato cyst nematodes in India. Objectives were to collect potato cyst nematodes from potato growing states of India, to develop a lamb-based diagnostic protocol for PCN, and to determine the genetic variability of the populations of PCN. The species were confirmed using species-specific PCR amplification. For development of the lamb-based detection protocol, a set of 5 PCN primers were designed based on the ITS regions of the genomic DNA. A PCN LAMP assay was developed for identification of PCN. The LAMP reaction could be completed within 75 minutes at 68 degrees centigrade, followed by termination at 85 degrees centigrade for 7 minutes. This assay was found highly sensitive and specific for identification of globular osteogenesis. The LAMP assay can be used at field, quarantine, and other infested plant and soil materials for identification of potato cyst nematodes. PCN populations were further characterized by established SSR markers. For globular osteogenesis, 7 SSR markers, and for Globulidara pallida, 5 SSR markers were used for genotyping of PCN populations. The study demonstrated that globular osteogenesis and pallida populations from Himachal Pradesh, Uttarakhand, and Jammu and Kashmir were genetically similar. The Indian population of PCNs were found highly similar with the Bolivian populations, and this suggests that the PCN possibly spread to India from South America, particularly from Bolivia. Gagandeep Singh, MSc student, worked on detection of entomopathogenic nematode by loop-mediated isothermal amplification. Objectives were to develop and validate loop-mediated isothermal amplification protocol for detection of heterohybrid species and Stenonema species from total soil DNA. For rapid detection of EPN from soil, EPN LAMP was designed, and the primer designed using ITS and 18S ribosomal DNA of genomic DNA, and the reactions could be completed in 60 minutes at 66 degrees centigrade and 68 degrees centigrade respectively, followed by termination at 85 degrees centigrade for 5 minutes. The lamp designed here could detect even single EPN juveniles in 250 mg soil and up to picogram quantity of DNA in soil. It would be of immense utility for efficient detection and identification of EPN in large-scale surveys. Bosant Deswal, MSc student, worked under my supervision on characterization of Gypinema longidorus and Paratychotus population with the objectives collection and identification of population from IRI farm, then morphological and molecular characterization of all 3 populations. He collected sample from 18 crops and identified Gypinema bacilli from haze plant, Longidorus species and Paratychotus species from rhizosphere of citrus orchard from IRI horticulture farm. A population of Gypinema was recovered from the rhizosphere of citrus from IRI farm, and it was morphologically and molecularly characterized and identified as Gypinema bacilli. Another population of Longidorus was recovered from the haze plant from IRI, and population was morphologically and molecularly characterized and identified as Longidorus species Edward et al. 1964. A population of Paratigoras was recovered from the rhizosphere of citrus from IRI farm, and this population was characterized by morphology and molecular study. However, the species appears to be new, therefore additional marker gene sequence are essential for confirmation and designation of the new species. Viresh AM, MSc student, worked on molecular characterization and expression profiling analysis of Photorhabdus toxin receptor genes from Galleria melanola. Objectives were RNA isolation, cloning, characterization of receptor genes, then RNA and PCR-based expression profiling of candidate receptors in Galleria melanola treated with TCAB toxin. A mid-cut receptor gene cadherin was molecularly characterized from Galleria melanola. The coding sequence of GMCD consists of extracellular, transmembrane, and intracellular domains. 14 cadherin repeats predicted in the extracellular domain. The Lepidopteran CD sequence was found highly conserved and distinct from the other insect clades. Expression studies of GMCD revealed that it was greatly expressed in 4th instar larval stages and in midgut tissues as compared to the other body parts, and silencing of the gene reduced Galleria melanola susceptibility to the TCAB toxin. This study also characterized GPI-anchored protein. Both GM-ALP and GM-APN function as the receptor proteins, and both the proteins were greatly expressed in the midgut epithelial cells of 4th and 5th instar larvae. This study suggests GM-CAD as one of the functional receptor in Photorhabdus acasti TCAB induced toxicity in Galleria melanola. It is also assumed that this toxin mimics the crite intoxication mechanism in Galleria melanola, and this forms the basis of future research to the insect gut receptor interaction with Photorhabdus toxins. Because PhD student worked on development of application technology of Heterorhabditis indica and its post-application adaptation under open field and protected cultivation, the objectives were to standardize the delivery system of Heterorhabditis indica using different spray equipments, to standardize the release of Heterorhabditis indica via deep irrigation, and to study the post-application survival and persistence of H. indica under protected and open field. The significant findings of this study: different types of nozzle, flat fan, flat jet, hollow cone, and Duromist nozzle up to 60 to 80 psi had little effect on survival and infectivity of H. indica infective juveniles. Different adjuvants like Twin 20, Twin 80 were found compatible with the infective juvenile. Soluble fertilizers were also found compatible with H. indica and TAN mixes. Then deep irrigation pressure up to 400 kilopascal had little effect on H. indica. Then migration, horizontal and vertical from the point of release, was also correlated with the time and distance. Then survival of the infected juvenile up to 1 hour was found when applied early morning on tomato in protected system and on soybean in open system. Then infective juvenile can kill up to 40% of Spodoptera larvae infesting soybean. Virender Kumar, MSc student, worked on RNA effect of the sex and larval development genes on root knot nematode development and multiplication. Objectives were to identify genes playing functional role in sex and larval development of Merodogon incognita, and to express the double-stranded RNA construct of this gene in Arabidopsis and study their effect on root knot nematode development and multiplication. By in silico analysis, TRA1 and BC81, these two genes were identified, primers were designed and amplified the target region. Agrovector mediated transformation was done and confirmed by PCR reactions, and the double-stranded RNA construct was developed, and the transgenic events were further confirmed by PCR reactions. The effect of gene silencing on root knot nematode was assessed. There was significant reduction of the female gall and egg per egg mass in the transgenic event. Thus, the silencing of TRA1 and BC81 interferes sex and larval development of root knot nematode. Thus, these two genes could be the potential target for nematode management through RNA approach. Opsorian, MSc student, worked on evaluation of wild brinjal germplasm against root knot nematode Merodogon incognita. Objectives were to study host response of different wild species of brinjals against root knot nematode, to study development of Merodogon incognita in resistant and susceptible succession of brinjal. The wild species of brinjal were included and number of accessions were evaluated. Significant findings of this study are: in Solanum khasianum, most of the accessions were found susceptible to Merodogon incognita. Solanum torban was identified as best source of resistance to Merodogon incognita. In plumeriget studies, the nematode penetrated in the resistant accessions of Solanum torban, but further development was delayed. Manoranjan Das, PhD student, worked on molecular dissection of rice, Melodogony graminicola interactions, effectors and targets. The objective of his studies were whole genome and transcriptome sequencing of selected rice and susceptible activation tagged mutant lines, identification and characterization of Melodogony graminicola effectors and functional validation of key effectors involved in nematode rice interaction, then identification of cognate rice protein interacting with selected nematode effectors. Whole genome sequencing of activation tagged nematode resistant lines were compared to their parent background. A large number of SNPs and indel variants were identified. SNP and indel variants were identified in genes related to transcription factor, metabolism, resistant traits, and some susceptible genes. RNA sequencing of the line number 9 were compared to the wild type JBT36x14. 674 differentially expressed genes were found after inoculation of 24 hours, and it was found that L9 was incompatible, and this was primarily responsible for early recognitions of the nematode by activation of all associated receptor kinase, RLKs, and several other signaling genes, and the nematode induced the triggered immunity in the plant mediated by phytoalexins and PR proteins. In this study, number of secretory proteins and the orthologous parasitic effector sequence were identified from the Melodogony graminicola genome. 13 Melodogony genes were functionally evaluated by in vitro RNA assay, and it was found that nematode penetration and multiplication were reduced. This study also identified the cognate rice protein interacting with nematode secretor effectors CLE genes by Is2 hybrid approach. 4 rice proteins were found interacting with the CLE genes of Melodogony graminicola, and CLE protein were found interacting to be involved in apoptosis, transcription, translation, modulation of steroid hormone biosynthesis in gene, and these proteins are found involved in the suppression of plant defense response and modulations of basic cellular physiology. Our student received awards, prizes, Fulbright Fellowship, travel grant for participation of international conference, thesis competition awards, first, second and third prize, poster competition, first and second prize. Our faculty and student published 25 research papers during 2021. The PG activities in the division involve professor-student meeting, MSc PhD student Vivavushi, and conducting several examinations through online and offline. Thank you for your patient listening.
Thank you very much, Professor M.R. Khan. Now we will move to next presentation, last presentation of this school, that is plant pathology. Professor B.K. Barnwal will make presentation. Thank you.
Very good morning. I'm here to present before you the most significant research achievements of postgraduate students in the division of plant pathology. The mandates of the division include to conduct basic and applied research, to serve as center for academic excellence, to provide national leadership in plant pathological research. The division has a faculty strength of 26, and 2 members of the division superannuated. One of our faculty members unfortunately left this world due to COVID-19. Here are the details of courses offered in the academic session 21-22. There are 4 major sections in the division of plant pathology: fungal pathology, mycology, virology, and bacteriology. In 2021-22, there are total 95 students with the faculty student ratio of 1:3.8, which increased from the faculty ratio of 1:1.75 in 2019. There are 10 MSc degree recipients whose names and their thesis title along with the name of chairpersons have been listed here. Similarly, there are 6 PhD degree recipients, and their names and title of thesis along with the name of chairpersons are given here. There are 4 major theme areas in the research in the division of plant pathology. In the theme area 1, diagnostics and pathogen characterization, Mr. N. Bhupati worked on morphomolecular characterization of Ganoderma species collected from Erica nut. he he grouped these ericanut infecting Ganoderma species in two major groups, stipitate and sessile, and he also characterized these Ganoderma species based on internal transcribed spacer regions as well as phylogenetic relationship. Ganoderma arbiforme was reported for the first time on ericanut, and these Ganoderma were also evaluated for their volatile compounds. Mr. Baswaraj worked on morphological, pathological, and genetic variability among Alternaria brassicae isolates infecting Indian mustard, and he also evaluated effect of crude toxins on symptom developments in Brassica gensea. He found significant variation in cultural and morphological characters, and based on DNA fingerprinting generated by ISSR markers, he grouped 20 isolates of Alternaria brassicae into three main clusters, and during screening, he did not find any genotype showing resistance to Alternaria brassicae. He also showed that 100 and 1000 ppm crude toxins from Alternaria brassicae isolates induced symptoms on all the genotypes. Mr. P.V. Dinesh Kumar worked on identification and molecular characterization of viruses and phytoplasma associated with the carrot. The carrot plants in the field showed little leaves, leaf curl, chlorosis, witch's broom, and phyllodes symptoms. When he subjected some of these plants in electron microscopy, he found the flexuous virus particle which could be decorated with the antibodies of a potyvirus species. Based on sequencing of amplified product of potyvirus degenerate primers, he identified the virus as carrot thin leaf virus Delhi isolate, which is a first report in the carrot from India. He also found the association of geminate virus particle, and based on the RCA product sequencing, he found the association of cherry tomato leaf curl virus as well as croton yellow vein mosaic alpha satellite. While characterizing phytoplasma associated with carrot, he found the association of 16SR2D subgroup of phytoplasma not only with the carrot crop but also with the vectors and weed species. Mr. Manish also worked on multi-locus gene-specific characterization of phytoplasma associated with bamboo species and its natural reservoirs collected from different parts of India, particularly in northern part as well as southern parts. And here are the symptoms which he found on different bamboo species as well as weed species growing around the bamboo plantations. He characterized the phytoplasma affecting different bamboo species and showed that these phytoplasma belong to different group or subgroups such as 16SR1B, 16SR2D, and 16SR2C. He also characterized phytoplasma from the weeds and vectors, and they belong to almost similar group of and subgroup of Rajinder Jatos worked on characterization, diversity, and PCR-based diagnostics of bacterial stalk rot of maize caused by Dickia G. And he revealed the genetic diversity in 17 isolates of this pathogen, and that was assessed by repetitive-based PCR. In the theme area, host-pathogen interaction, Jagmohan Singh deciphered the Chaetomium globosum induced signaling network in tomato against early blight. Among delivery methods, he found that seed treatment supplemented with single or double drenching with the Chaetomium globosum provided better plant growth and physiological parameter. He showed that their activation of salicylic acid and jasmonic mediated signaling transduction was there in Chaetomium globosum treated tomato plants, indicating involvement of both ISR and SAR mechanism of induced defense. In tritrophic interaction studies, he showed that plant hormone signaling was very active in biocontrol treated plant, and they increased manifold on counter inoculation with the pathogen. In his study, he found that there was significant increase in 30 differentially expressed genes in tomato plant treated with Chaetomium globosum. Kuleshwar Prasad Sahu worked on structural and functional analysis of rice phylospheric bacteria for their antimicrobial properties and defense elicitation against blast disease. He did metagenomic analysis for 2 rice genotypes in 2 contrasting geographical locations and found that the Pantova and Bacillus were as most dominant bacterial genus by metagenomic analysis as well as cultural population analysis. Using dual culture and volatile confrontation as well as seed priming followed by in planta assay for inhibition of blast disease, he identified promising bacteria and showed that there was activation of MAM-triggered immunity pathway genes in rice. GCMS-based profiling of bacterial volatile resulted in the identification of compounds with antifungal properties. In the theme area pathogenomics, Mr. East Prakash studied population biology and did genetic analysis of blast pathogen Magna portha grisia infecting pearl millet. In total, he collected 20 blast isolates from all major pearl millet growing areas, and he found that the sporulation ability of this pathogen correlated with the color of the mycelial growth and virulent nature of the isolates. He also showed that there was a deletion of 4 nucleotide at the 36 to 39 position of PFK gene in the sequences of pearl millet blast isolates. He also studied relative expression analysis of pearl millet blast hypothetical virulence loci during blast pathogen host interaction and showed that these hypothetical virulence genes of pearl millet infecting Magna porthae, infecting the pearl millet were found to be progressively increasing in a temporal manner in susceptible genotypes as compared to resistant line. Mr. Firoz Mandal did evaluation of resistance behavior of TY genes containing donor lines of tomato and indexed naturally occurring bigomoviruses infecting them. He found that 5 genotypes, all containing TY2 and TY3 genes, were found highly resistant. He also showed that there are 6 different bigomoviruses which are found in different combinations in the field condition. He characterized a new bigomovirus species which was named as tomato leaf curl TY Pusa virus. In addition to that, he also recorded for the first time tomato leaf curl Karnataka virus from northern part of India. Mr. Kavi Siddharthan worked on identification of robust approaches for virome analysis for understanding the seasonal dynamics in virome of grape vines exhibiting leaf roll syndrome. He found that the whole transcriptome and mRNA-based virome analysis provided better genome reconstruction of viruses and identified more number of viruses in comparison to small RNA-based virome analysis. He developed a virome analysis pipeline, and using that standardized virome analysis pipeline, he discovered large number of novel viruses in different crops and published papers in Gene, Archives of Virology and Plant Gene as well as Journal of General Plant Pathology. Using this mRNA-based virome analysis, he determined the copy number of complete or partial genome of viruses and viroids from 2 varieties from the grape growing region in Maharashtra, that is particularly from Pune, and showed that the copy number was highest in the month of September as well as in December, but it was quite low in the month of December. Sorry, the highest number of copy number of viruses and viroids was in month of September and March, and it was least in the month of December, and that was also confirmed through real-time PCR. In the theme area of plant disease management, Mr. Babu genotyped Fusarium graminearum isolates for trichothiocin toxins and identified head scab resistant sources in wheat. He determined pathogenic variation among 27 Fusarium graminearum isolates and showed that all these isolates belong to 15 add-on type based on amplification of toxin-specific primers. He found that one genotype DVW302 and 9 indigenous germplasm lines were moderately resistant out of 344 wheat genotype tested against this pathogen. Mr. Mukesh Kumar worked on structural and functional analysis of rice endophytic microbiome for antimicrobial compounds against rice blast caused by Magnoporthy oryzae. He did microbiological investigation in 3 geographical locations, resulting in a total of 283 endophytic bacterial isolates. And all bacterial isolates were phenotypically characterized, DNA fingerprint, species identified, and preserved. He also studied the effect of bacterial cell culture and native bacterial volatile organic compounds on expression of different genes in rice. And in general, he found that volatile treated seedlings showed up regulation of all tested genes as compared to live cells. Mr. Muhammad A. Ayum worked on genotyping virulence analysis of Tillecia indica and identification of kernel bunt resistance sources. He studied for the first time to SNPs linked virulence to Tillecia indica for the first time, and he also identified virulence genes based on pathogen host interaction database. Most of the selected candidates' virulence genes were highly upregulated in susceptible variety in compared to resistant varieties. He also showed that the 31 genotypes were resistant out of 36 genotypes when tested against this pathogen. Ms. Sabya Singh worked on identification of putative mild cross-protecting strains of citrus trichia virus for management of severe decline disease in citrus. She characterized a large number of citrus trichia virus from the northeast India and found that 6 of these isolates showed recombination events. And based on the codon usage variation using code protein gene of CTV isolates, she demonstrated that 3 isolates had characteristics of putative mild strain, which can be used for cross protection against the severe strain of citrus trichia virus. Mr. Karyappa Chaudhkar worked on microbial antagonists in biological control of wheat powdery mildew pathogen. He found that Bacillus subtilis provided the best protection against powdery mildew pathogen in the wheat, and he also showed that there was maximum increase in growth parameters and defense related enzymes when this biological control agent was used in the wheat. Ms. Sweta Mishram studied the mechanism of disease resistance in maize during maize leaf blight infection, and she showed that there was a high wax deposition trichome and smaller stomatal size, which offered hindrance to pathogen for entry and proliferation. She showed that the chlorophyll content was high in resistant inbred line compared to susceptible lines, and there was upregulation of pheophytinase gene, the key gene of chlorophyll degradation pathway, in the CM119, indicating that there was higher degradation of chlorophyll due to maize leaf blight disease. She also showed enhanced expression of PAL and beta-1,3-glucanates in the resistant line of maize upon infection with the blight, this Medes leaf blight pathogen. She also showed in the real-time PCR expression of few phytinase gene in susceptible as well as resistant lines. So these are the honors and positions secured by student. Miss Asarani Patel got the Bairek Sitare Gaiti Award, and in this award she received 15 lakh rupees for conducting research in her area of microbiome study. Sweta Mesram received Best PhD Thesis Award as well as Best Research Scholar. Darshan, Pradeep, Kabi Siddharthan were posted as Scientist B in Indian Council of Forestry Research, and Mr. Jagmohan was placed as Subject Matter Scientist in Plant Protection. So these are the awards and honors conferred on scientists. Rashmi Agarwal got T.S. Thind Award by Indian Society of Plant Pathology, Ludhiana. She also received P.V. Munkar Award of IPS. Myself got the most coveted National Professor, ICR National Professorship, and Robin Gogoi got SR Memorial Award. Similarly, Vikas Mandal and MS Saran got NAS Fellowship in 2022 and 2021. In total, there were 133 publications during 2021-22, and many of these papers were published in high-rated journals such as Cells, Journal of Fungi, Frontiers in Plant Science, Post-Harvest Biology and Technology, Frontiers in Microbiology, Scientific Report, Plant Disease, and so on. Thank you, one and all.
Thank you very much, sir, for nice presentation. So, Chairman, sir, with this, we have completed all 4 presentations. And now I invite Chairman, sir, for kind remarks.
Well, at the outset, I wish to congratulate IARI for the 6th convocation to be held very soon. I'm not really very sure, probably Dr. Aki Singh or Rashmi may probably correct me. This is one of the very unique platforms where the postgraduate research work is being highlighted as a part of the conversation. I'm not sure whether it is anywhere else existing in our system, including SAUs and so on and so forth. And if my judgment or if my statement is right, then I would say it's an additional feather in the professional cap of IARI. And I must compliment both the director, the dean, who are actively involved in academics, that such an exercise is being made to bring sharper focus, the quantum and the quality of work being done by the postgraduate. And this also speaks volumes about the level of competence which the major advisors or the supervisors or the faculty members are imparting upon the younger generation. So congratulations to each one of them. I am also very happy that it has been catalogued their achievements, list of publications, honors, etc., etc., etc. And I would say again, it has this time I could say still better improvement than what I saw about 2 years back, if I recall correctly. I could also see that the thematic areas which the different divisions have made have been well placed. But then I wish to also take the opportunity Dr. Singh to share my
कन्वर्सेशन चल रहा है तो हर दिन 20-20 मिनट ज्वाइन कर लेता हूँ बस ज्वाइन कर लेता हूँ
Please meet somewhere, Prakash. Please go ahead, sir. You are muted, sir. Muted, sir. Please unmute yourself.
Kindly unmute, sir. Doctor.
Okay. So, nevertheless, I do recall last time also that climate change, except probably, if I remember correctly, the division of entomology has some studies made as one of the thematic area in climate. And yet I could not see much about it in other divisions. And I must emphasize, Dr. Singh, Dr. Rashmi, and everyone else, and all professors who are attending this, that climate change is bound to rattle the apple cart of agriculture in the years to come. We still don't seem to be getting sensitized about the issue, particularly in the domain of plant protection, where even the baseline information is not available. I would say, realizing that at PhD level, you don't have many years to go because climate change is an ongoing process. But these students could be exposed to different dimensions and generate the baseline information on this domain. This could be very critical in the years to come. Nevertheless, it is heartening to see that the students are in fact adopting the techniques of precision in their study, and that is very, very gratifying. In the division of plant pathology, mycology still happens to be at the lowest priority. Now, I am not sure. Lot of discussions have taken place on national platforms, and the taxonomy of particularly fungi and bacteria, they are still getting low priority. And time will not be very far when even the identification of these organisms will be a very tough job. Though we do have techniques available, molecular taxonomy, for that matter, and so on and so forth. I don't have to. Well, with this, I'm sure we have already crossed the boundary of time limit. Nevertheless, it has been my pleasure always to have an interaction. It was a learning experience. Every moment is my learning experience. And I wish you best of luck and success in pursuit of your nurtured and nourished missions of professional career. God bless you all. Thank you very much.
Thank you, sir.
Thank you so much, sir. Thank you so much for your kind words. And we will try to adopt the two priority areas that you have mentioned about the fungal taxonomy and mycology that we could identify. Certainly, we have our Indian type culture collection that is being maintained regularly, updated also, and we have full-fledged scientists associated with that. But we will certainly strengthen.
Yes.
Insect taxonomy, we are doing very well, and as a matter of fact, one of the students presented the work yesterday on the variation in moth species in IRI campus and Delhi, and the work was very well appreciated by all the judging committee members. So we will try to take your suggestions in view and act on it. Thank you, sir.
Yes, sir. Agreed here, sir. I also agree that we have only now another mycologist will be superannuating, sir, in this year. So definitely we will have to have more taxonomy, especially in fungal taxonomy, sir. We will look into it, sir.
Yeah.
Thank you.
Thank you. Thank you, madam, and thank you, director, sir. Before moving to this next school, I take this opportunity
Sorry,
to
sorry, Dr. Datta, for a moment. Dr.
Sir,
Datta, for a moment.
sir.
I see Dr. Gore here. I would appreciate if a couple of minutes could be given to him.
Yes, Dr. H.S.
Yes,
Gore,
to
yes,
give his
yes.
own observations. He is a well-known plant protectionist in general and nematologist in particular. I think let us request him to give some little briefing about it.
Hello. Yes. Gore, sir.
We would like to have your comments, sir. Dr. H.S. Gore, former Dean IRI. Sir, Dr. Gore's observations on the presentation of crop protection, sir. In particular, and nematology, particularly nematology and in general crop protection. Sir, Dr. Gore, sir.
Yeah, unmute
sir, you are unmute kar lijiye, sir.
Hello. In fact, I was glad to see the work that has been done in all the divisions of the plant protection school and other schools also. I've been continuously watching. And it's a pleasure that to see the standards of research are going up, quality of publications is going up, and nematology in particular also a lot of development has taken place, and a good number of students, 5 MSc and 3 PhD, have been present. This number is increasing. Good amount of work has been done. Only thing is more work is going towards molecular aspects and biochemical aspects. Nematology still needs a lot of applied research. Nematology still has not gone to the farmers too much actually. Even there are people, even in plant protection schools, other departments, people who are not aware of nematology. Work is also necessary on multi-trophic interactions, particularly in disease complexes, where nematodes play a much bigger role. Fungal disease complex, bacterial disease complex. So there is a lot of necessary work to be done there also. I'm not saying that this work that has been done is not good. It's all very, very good work that has been done. But in future, I think if some problems can be given to students also on disease complexes and applied nematology to a greater extent, that can be of great help. And also development of specific chemical molecules, agricultural chemicals particularly, they need to find out nematicidal molecules, because nematicides are in big paucity in the country actually as such. There are hardly any nematicides except 2 molecules have come now, which are not very popular. So there also some development is required, molecular development or identification of or identifying some nematicidal products where this has to be done. I must compliment the work. Actually, I cannot say that this is the only work being done, because this is only students' work which has been presented. Maybe other people are working in that direction. So it's a good thing that developments are taking place, and I wish all the best to the students who have done so good work, and I wish they can continue this in future and wherever they go in different institutions, and a lot of new developments are expected. Thank you.
बना रहा हूं मैं लगातार 3-4 राउंड ऊपर नीचे
Please mute
फोन आ गया किसी फोन से
please
इसकी जरूरत नहीं क्योंकि मैं डायरेक्ट इंटरव्यू कर रहा हूं
who is there please
Mansingh.
Okay. So thank you very much, sir. Thank you, God sir, for your kind appreciation and critical comments. So before moving to the next session, I take this opportunity to express profound sense of gratitude to Dr. Bhushan Jalali ji, a very senior pathologist having vast experience, national and international exposure, for gracing this occasion as chairperson and also sharing valuable thoughts and giving critical comments. I am sure this will help us in shaping our research in our future endeavor. Thank you very much, sir, for your kind blessing. Thank you, sir.
Thank you.
Now, I request our Dean, Madam.
Yeah.
Yeah.
I will have to, sir, Next session
we will have to
sir.
take some 5 minutes break. Dr. S.K. Chaudhary is in meeting with Director General. I am also in the same meeting simultaneously. So the meeting is going to be over shortly, and then he will join.
Okay.
So
Okay,
we can take a break for 5 minutes and then return.
We
Okay,
let
sir.
us be keep joining, like we should not leave.
Just be joining.
And then
he will join any moment.
Okay, sir. We are joining. Anything other than this, sir?
प्रोफेसर गौड़ नमस्ते।
नमस्ते नमस्ते मान सिंह जी,
कैसे हैं सर?
वेरी गुड, थैंक यू। So ठीक है, बिल्कुल। इनफैक्ट lot of knowledge coming in and I'm trying to grab. I don't know where I take it.
Good, sir.
But at least I try to.
You are still active in academia.
Yeah,
तो अच्छा है, students को आप interact करते रहते हैं.
I was in fact glad to see Professor Jalali so active.
Yeah.
He is much more senior to me.
How is your health, by the way?
I'm perfectly fine.
Good.
With your good wishes. And if I see IRA anywhere, I become younger.
You become energetic. I mean, you get a lot of energy. A lot of energy is transferred to you.
IRA means a lot of vigor comes from there.
And a lot of energy, gets radiated through our director, Dr. Aki Singh.
Oh yes, certainly. That's why I'm joining.
He emits energy always.
Oh yes.
Dr. has completely transformed Sarda University in agriculture. Yes.
Yeah, it's a mini IRA.
Yeah, you have created that.
It's a kind of just so कहते हैं प्रोटोटाइप वो होता है ना, जो मिनिएचर बनाते हैं, कहते हैं कि बड़ी कार का छोटा मॉडल होता है तो टॉयज में बिकता है ना, it's a kind of miniature IRA here. So we have similar pattern we are following. We are not in the same magnitude, but we are going in different things.
You are Tata's Nano.
Maybe Nano तो बड़ी है, फिर भी मैं तो बहुत छोटा बता रहा हूँ. But we are doing some work in different directions. Particularly, we are diversified into different aspects. I'm trying to go for high value agriculture. We have 3 different kinds of hydroponic systems now put up. There is a world-class tissue culture and hydroponic lab which has been set up.
Okay.
We are also producing stevia. We are producing cordyceps mushroom. Cordyceps mushroom. We have pleurotus mushroom center has been established, and tissue culture of medicinal plants we are doing. So these things we have now created actually, and so my intention is to rooftop gardening, particularly rooftop horticulture and rooftop peri-urban systems, urban beekeeping and all that. I'm trying to go towards peri-urban aspects and also getting more and more income from small area, and also training people. We are trying to organize trainings for this youth of the area on technology like say pleurotus mushroom, what is this, oyster mushroom and all that. It's so easy to produce, that kind of system. And also seed production we have started. Wheat seed we have produced last time. We distributed to a lot of farmers of the area, and we are receiving good reports from them. I got seed from Karnal, different varieties, 187, 222 and all. This time again we are producing, and the farmers are giving good report. So I am just only extending the technology of ICR and ARI and giving to public actually. So I think still, and we have faculty of around 23 now actually in core faculty, plus 13, 14 other people from other schools also teach. So overall more than 30 people are teaching BSc agriculture, and MSc agronomy we have started. And PhD also we have started in agriculture. So good work is going on. Let us see. But this is a beginning. I think I cannot compare with IRA certainly, but and the faculty which you have taken is very good faculty we have taken, quality faculty, trained abroad or in different institutions of India, and also some are from ICR system itself or ICUs. They are doing very good. IIT Kharagpur faculty also have somebody from Finland, done PhD in Finland, that are also here. We are also doing some work on the ozone pollution, environmental science, so ozone and effect on plants, particularly concentrations in this area. Multifarious kind of things. But Dr. Aki Singh, one thing is good to see here, that Mansingh, that in fact, there are almost 700 people joining at this time. Which I think is a better way. We used to have difficulty in filling the auditorium, where only 200, 300 people were there. Seats, even those 300 seats are not filled. We're asking people to come, all the time. But at least here now we have more than around 700, more than 771 I saw at one point. So many people are able to join and take part in it. Maybe the gift of COVID, I think.
And the blessing of technology.
Yeah, the blessing of technology and the lesson taught by COVID to us.
Absolutely.
COVID told us that you are wasting time. You better do this way. Good.
You can also have simultaneously more meetings, as our director sir is also doing, and many times we do.
Yeah, this is what I have been doing actually, and we have been attending to my work and also like this,
sitting in office and doing other jobs also.
Yeah.
Back-to-back meeting.
Yeah.
Yes, agricultural microbiology, we have Professor Dolly Wataldhar here. Actually, she is doing agricultural microbiology. And
she's
doing in spirulina, she is doing cordyceps is also done by her actually. It's her baby. And so we are doing, high
value medicinal mushroom.
High value mushrooms. Yeah. I can show you the product with me right now here.
Oh, great. So culturing is very difficult. Is it being cultured?
It's very difficult, but we learned actually, we perfected. We have perfected our technology now.
Very good.
And
excellent.
So it's good actually. And this much may be costing around 20,000-30,000 rupees.
Yes, sir. It's very costly. Because naturally, if you collect, it's very difficult. You don't get them. Only high Himalayan ranges you get under natural conditions.
No, but we are doing
the forests.
Yeah, we are doing a different species. Actually, we are going for militaries, which is easier to run.
Easier to run.
Sinensis is difficult. Militaries can be done in. We are doing in a vegetarian condition, particularly here.
Okay. That is why people can
produce it.
Eat
Yeah. So this is a vegetarian mushroom. Cordyceps. The microbiology is being attended to, and also waste management we are doing, vermiculture, other things we are also doing. And blue-green algae, of course.
The site is also.
When Dolly is here, that means blue-green algae is here. We have a collaboration with this microbial, Saxena is there. So we had MOU with them, and we are doing some collaborative work with them. And that's why I was asking for a collaboration
Yes,
with IRA itself. Actually, IRA also we could do a lot of things.
yeah,
So this
just
is
still under consideration only, sir. We will see.
Yeah. So there's much, there's something easier here because some things can be done easier in the private system than in the government. I tell you this thing, the kind of work I could do in just 4 years, do this thing, would not have been possible in the ICR system. With that officialdom, no? We don't have that official. Today, I think this should be done. I get it done next day. So that's the possibility. That is possible here. And the kind of freedom this management gives, the Sharda University, I must salute the management here. They really give that kind of freedom, autonomy. That you are in charge of this thing, do this thing, that's all, finish.
Yeah.
And whatever I do, they don't question it. They say, okay, this is required. I justify to them. Once they're convinced, go ahead. Nothing. And then new trials, new things, you can try anything.
Wonderful.
Dr. Chaudhary has joined Dr. Asmi.
Yes, sir. Yes, sir.
We can start now.
Okay, sir. Okay, friends, we are going to begin now the third session of the 60th Convocation Week program of IRI. And I profusely welcome our chairperson of this session, which is on the School of Natural Resource Management, Dr. S.K. Chaudhary, who is Deputy Director General NRM from ICR. Sir, we wholeheartedly welcome you. And we know that you are so busy and still you spared the time and joined this particular session today. So on behalf of Director and on behalf of the faculty of IRI, I welcome you, sir. And this is a session wherein the professors of disciplines under the School of Natural Resource Management, which is the largest school here at IRI, they will be giving presentations of the significant educational achievements done in their respective disciplines. So this is pre-recorded, sir, and there are 7 disciplines under the School of NRM, sir. These are agriculture engineering, agricultural physics, agronomy, environmental sciences, microbiology, soil science and agriculture chemistry, water science and technology. And there are total 76 theses. 76 students will be awarded degree during this particular convocation. So some MSc and some PhDs. So total MScs are 56 and PhDs are 22 theses. So the professors of these respective disciplines, they will be giving their presentations. So with this before very brief introduction, I would like to now hand over to Director Dr. Aki Singh to formally introduce the chairman.
Thank you, Dr. Rasmi, and I'm sorry for dragging Dr. Chaudhary from one very important meeting which is still going on to this convocation meeting, sir, but this is also extremely important and we are so happy that you have kindly agreed to chair this session. Friends, Dr. Chaudhary did his Bachelor in Agriculture and Master's in Agriculture from JNKV Jabalpur and PhD from MPKV Rahuri. And he served as a researcher and head Soil and Crop Management Division at Central Soil Sanitary Research Institute for a long period. And his major focus of research has been on dynamics of irrigation-induced land degradation, soil hydraulic properties under different quality of water, influence of poor and marginal quality water on soil properties and plant growth. He also served as the Assistant Director General Soil Water Management, and you all know that currently he is serving as the Deputy Director General Natural Resource Management at the Council. His initiative on a mega project involving multi-institutions on precision agriculture, where IRI is leading the project, is primarily his brainchild, and this is such an important project that this is going to provide a complete transformation to farming system, be it agriculture, animals, fisheries. Dr. Chaudhary is recognized for his outstanding work with the Rafi Ahmad Kidwai Award. We are so happy to have you, sir, on the to chair this particular session on natural resource management, where the significant research finding of the students who could not compete for the best student medal, but their work is no less than the other students, and their work is compiled and will be presented by the professors of respective discipline. And we will hear your conclusion, and also your suggestions are guiding the future research program in this discipline. I welcome you once again. Thank you very much.
Thank you very much, sir, Director sir. So, as you know, in NRM division, NRM school, we mainly deal with the sustainable utilization of major natural resources such as land, soil, water, air, which is of utmost importance for our very survival, prosperity, as well as posterity. And as Dean ma'am already made mention, this is one of the largest school of IRI and comprises of as many as 7 disciplines. So with the kind permission of Chairman sir, may we take a first presentation?
Yeah, please go ahead.
Okay, sir. Thank you, sir. So first presentation will be from Agriculture Engineering, and Professor Dr. Singh will make presentation.
Honorable Director IARI, Chairperson of the session, Dean and Joint Director Education, Project Director WTC, Head of the Divisions, Professors, Scientists and Students. I am going to present the significant educational achievements of the Division of Agricultural Engineering for the year 2021-22. Agricultural Engineering Division has 3 sub-disciplines, namely the Farm Power and Equipment, Soil and Water Conservation Engineering, and Agricultural Structures and Process Engineering. Division had 29 faculty members at IARI and 46 at CIA Bhopal. There are 140 students on roll. 22 students will be receiving their degree in this convocation. Externally funded projects are the great source to our students. We have 15 externally funded projects with the budget allocation of 566 lakhs. Last year, the students' research was carried out under 6 themes, namely the resource conservation technology, precision farm equipment, climate change impacts and integrated water resources management, precision irrigation, solar-powered farm equipment and storage structures, and post-harvest technology for preservation and storage. There was one thesis under the resource conservation technologies. A battery-operated groundnut decorticator was designed, developed and evaluated. The output capacity of developed battery operated groundnut decorticator was more than the traditional method of decortication, and the efficiency was highest at 70 strokes per minute. There were 9 theses under the theme Precision Farm Equipment. MTech students developed a smart device for measurement of cutting energy, which was evaluated in the field for rice cultivars. Accuracy of device varied from 94 to 99%. Grape is an important crop of India. A student developed a sensor-based control volume sprayer for grape clusters. He measured the physical property of grape clusters from the research farm of the Division of Fruits and Horticulture Technology. Study included determination of design values of sensor, development of sprayer, and performance evaluation of developed sprayer for grape cluster. It was found that there could be saving of 30% in chemical use and 36% saving in operation time compared to the conventional dipping method. Decomposition of paddy straw is a big problem. A speed sensor-based microbial inoculum spraying system for combined harvester was designed and developed by a student for simultaneous application of microbial inoculum over rice residue during combined harvesting. The system was evaluated in the field and was found to be working well. Manual harvesting of pineapple is a time-consuming process. A MTech student designed and developed a pineapple harvester which was evaluated in Manipur. The developed harvester can dig 3 pineapple in 1 minute and has cost of about 4,500. A good amount of vegetables are damaged while transporting in the trailer due to vibration. A PhD student studied and quantified tractor-trailer vibration and developed a model to determine vibration during haulage. Several alternatives were tried and it was found that the corrugated fireboard boxes performed better than the plastic crates. Manual chop cutter are widely used machine for chopping fodder. In a survey, it was found that the design of chop cutter is not operator friendly. A PhD student studied the ergonomic aspect of chop cutter and modified the design. Property of the several fodder crops were determined and considered in modification. Based on the simulation studied, she found that the 3-blade with 66% reduction in throat size is ergonomically compatible. Biopesticides are increasing day by day. There are several biopesticides which cannot be sprayed uniformly with available sprayer. A PhD student at CII Bhopal developed a spraying protocol for biopesticides. He determined the effect of nozzle side, operating pressure, and pumping period on the viability of selected biopesticides. Performance of spraying system was evaluated in the field. It was found that the biopesticides should be sprayed at minimum operating pressure with larger orifice nozzle. Transplanting a pot seedling is a time-consuming and labor-intensive operation. An automatic tractor-drawn vegetable transplanter for pot seedlings was designed and developed by a PhD student at CII Bhopal. The performance of planter was evaluated with the tomato, brinjal, and chili seedlings. The field capacity of the transplanter was 0.13 hectare per hour with efficiency of 90%. This saved 95% of the labor and 93% of the time. Use of combined harvester in India is increasing day by day. Many times, operators have to work for long hours. Due to inappropriate location of control and seed design, operators are subjected to fatigue. A study on dimensions and location of controls and operators seed design was undertaken by a PhD student at CII Bhopal. Based on the study, he recommended the appropriate design for grip diameter, length of brake and clutch pedals, steering wheel diameter, and many other controls. The design parameters suggested by him matched with the anthropometry of about 96% operators. There were 5 theses under the theme of climate change, impacts, and integrated water resources management. The first study under this theme is on the crop water footprint assessment. As we know that the crop water footprint is relatively a new indicator for managing water resources and crop planning in a basin or a geographical unit. Earlier, one of our students has assessed the crop water footprints of Gomati and Betwa river basins. Last year, one of our students assessed the crop water footprints and the impact of climate change on crop water footprints for climate change scenarios under various RCPs. for Haryana state. 11 major crops cultivated in various districts of Haryana were included in the study. It was found that the virtual water content of rice was 2,946 meter cube per ton, and for the wheat it was 1,420 meter cube per ton. For sugarcane it was 3,285 meter cube per ton, and for the cotton it was 21,766 meter cube per ton. Blue crop water footprint accounted for 52% of the total crop water footprint. It was found that the crop water footprint of major crop would be increased by 4 to 30% as a result of climate change. Highest increase would be in maize and wheat. Groundwater levels are declining in several regions of India due to overexploitation for intensive agriculture. A study was undertaken by a PhD student to evaluate the impacts of water management technologies on irrigation efficiency and groundwater level using groundwater models. A study was conducted at Farmers Field in Muzaffarnagar district, which is the sugar belt of Uttar Pradesh. Based on the results obtained from the field experiment and simulation, he suggested management strategies for arresting water table decline. He found that the average irrigation efficiency for open channel and pipe irrigation system were 65% and 79%. Sugarcane-based farming system is not sustainable in the district, even though there was a positive impact of various management alternatives on groundwater behavior. Water table decline can only be arrested by bringing entire sugarcane area under drip irrigation and replacing 50% of the sugarcane area with maize and pulses. A MTech student conducted a study at Farmers Field in Gazipur district of Uttar Pradesh. He determined the on-farm irrigation efficiency and assessed the performance of surface and groundwater irrigation in terms of water productivity. He found that the average application efficiency in wheat for groundwater irrigation varied from 68 to 78%. The lowest was for electric operated tube well. The average application efficiency for canal irrigation was 56%. The average application efficiency in paddy irrigated by the groundwater varied from 66 to 77%, whereas for canal irrigation it was 53%. The average water productivity of wheat for diesel operated pumps was 1.1 kg per cubic meter, whereas for the electric operated it was 1.2 kg per cubic meter. Groundwater quality in coastal region is deteriorating due to overexploitation. These regions are facing problems of freshwater supply. A MTech student evaluated the groundwater level trends and delineated the area facing the problems of groundwater quality. Water quality parameters were collected and analyzed for the period of 2008 to 2018. He used Mann-Condell test for Piper diagram, Gibbs diagram, and USSL diagram in his study. He found that the groundwater level is declining rapidly in overexploited areas of the Visakhapatnam district. Sodium and chloride ions are the dominant hydrochemical phases in aquifer, and concentration of these ions have increased by 58% during 2008 and 2018. Presence of sodium and chloride ions in several areas shows that the groundwater salinity in several areas is increasing due to the mixing of seawater and freshwater. A MTech student assessed the water resource availability and crop water demand of Eastern Marathwada region of Maharashtra. She used crop wet and web models in her study. She found that the water resource availability is not enough to meet the demand in the Nanded district. Hence, prudent water management strategies have to be adopted for bridging the gap between demand and supply. There were two theses under the theme precision irrigation. Soilless cultivation is becoming very popular in semi-urban areas. To get the more yield and quality produce, application of required amount of water and fertilizer is important. In manual application, it is difficult to apply the required amount of water and nutrient as the volume of media on which crop is grown is limited for each plant. A PhD student developed a conditional controller-based fertigation system for water and nutrient management in soilless cultivation under greenhouse. Sensing system was developed to continuously monitor the water loss from the soilless grow bag due to transpiration. He optimized the fertigation schedule for cucumber and capsicum using the results obtained from the experiment. He found that the developed system was capable to detect the water loss, which helped in optimizing the fertigation schedule on real-time basis. He reported that the highest cucumber yield of 62 ton per hectare was obtained with 100% nutrient solution. Nowadays, a lot of emphasis is given on use of IoT in agriculture, particularly in precision irrigation. An IoT-based aeroponics monitoring and alert system was designed and developed by a MTech student. The alert system included automatic systems to monitor the EC, pH, temperature, nutrient concentration in solution, and humidity. Alert system sends notification on user's smartphone whenever any parameter is not in the range. User can start the system so that the required parameters are delivered to the plant. The good thing about the system is that the lag time is only 3 to 5 seconds, which helps to quickly control the parameter. There were 3 theses under the theme Solar Powered Farm Equipment and Storage Structures. Use of solar energy on Indian farms are increasing day by day. Government of India is giving major emphasis on promotion of solar energy in agriculture. The major problems vegetable growers in India are facing is the on-farm storage facility. Due to lack of storage facility, a huge amount of perishable vegetables are lost and farmers do not get better price. Our division has developed a solar refrigerated evaporatively cooled structures for safe vegetable storage. This is operated on off-grid and hybrid mode. MTech students evaluated the performance of this structure. He found that the leafy vegetable can be stored up to 14 days and it will be very useful for remote areas. The cooling efficiency of him was improved by 2.29 times in the off-grid hybrid mode. A PhD student evaluated the solar refrigerated evaporative cool structure for storing perishable agricultural commodities. She found that the leafy vegetable stored in the structure remained marketable for additional 3 to 5 days, and tomato could be stored for 32 days. Division has been trying different insulating materials used for evaporative cooled structure. A MTech student used hempcrete and a hemp fabric for designing and developing evaporatively cooled structure. He evaluated its performance for storing tomato and spinach. He reported that the inside temperature varied from 15 to 26 degrees Celsius under HECS and 17 to 28 degrees Celsius under AECS, when outside temperature was 38 to 40 degrees Celsius. There were 2 theses under the theme Post-Harvest Technologies for Preservation and Storage. Carrot is an important vegetable crop used as a salad in Indian houses. The surface decontamination of carrot is very common and needs to be properly cleaned before it is consumed. A PhD student at CII Bhopal developed a LED-based photodynamic inactivation system for inactivating microorganisms at the surface of carrot. She used incandescent lamp, laser, and light-emitting diode for this. Optimized value of the wavelength, frequency, power, and treatment time were 390 nm, 500 Hz, 10 watt, and 30 minutes respectively. With the developed LED-based PDI system, 10 carats can be decontaminated at a time and can be stored for 5 days. A PhD student at CII Bhopal developed a digital image processing based system for real-time monitoring of potato slices in hot air dryer. Dryer was equipped with an image acquiring system to visualize the color, texture, and morphological changes during drying. The system was evaluated for drying of potato slices. It was reported that the air dryer served the purpose and successfully dispersed the hot air throughout the dryer. This was all about the significant educational achievements of the students who will be receiving degrees in this convocation. We are very much satisfied with the performance of our students even during pandemic. Our students have worked almost in all the areas of agricultural engineering. They have addressed the issue of resource conservation, precision in agricultural operations, on-farm infrastructure, water resource management at regional and basin level, application of solar energy for safe storage of perishable produce, post-harvest management, and application of IoT in agriculture. I thank the students and their chairpersons for their continued support. Division has taken several new initiatives. We are in the process of establishing smart classrooms, which will be functional by the March. A YamTech program was initiated at IRI Jharkhand and NISM Baramati. We are going to expand our activities as per the recommendations of the latest BSMA committee. There were total 12 publications from the student thesis. Of this, one paper was published in more than 10 NAS rating journals. Our students were employed as assistant professor in SAUs, assistant engineer in state government department. The majority of our students opted for higher education. One of our students joined Washington State University for his PhD program. Our faculty members have received several awards and recognition as shown in this table. At the end, I thank students, faculty members, head of the divisions, PDWTC, Dean and Joint Director Education, and the Director IRI for their continued support and help.
Thank you, sir. Thank you very much, Professor Dr. Singh, for your nice presentation, as well as saving some of allocated time. Thank you, sir. So now, with kind permission of chair, we will move to next presentation. Next presentation will be from Agriculture Physics, and Professor P. Krishnan will make presentation.
Respected Chairman, dignitaries, scientific staff, students, ladies and gentlemen, I am going to present the significant educational achievement in the discipline of agricultural physics during the last 1 year. Agriculture physics is a unique subject in which research, teaching, and training is conducted by applying the principles and laws of physics to characterize the process of soil, crop, atmosphere continuum. It encompasses subdisciplines like plant biophysics, agriculture and meteorology, remote sensing and GIS, and soil physics. This year, 7 students are graduating in the discipline of agricultural physics, 2 PhD students and 5 MSc students. Their research work is categorized under the theme of biophysics and image processing, remote sensing in agriculture, soil physics, and crop simulation modeling. The thesis title of the PhD student, Mr. Kaushik Banerjee, is on thermal imaging for characterization of abiotic stresses in wheat under field condition. Mr. Kaushik has collected thermal and visible images from 10 wheat cultivars grown under abiotic stress conditions like nitrogen, moisture, temperature, salinity, and their combinations using a thermal camera, along with other plant biophysical stress parameters like LAI, NDVI, chlorophyll content, photosynthesis, respiration, etc. Using multivariate techniques, a new normalized stress tolerance index was developed for each stress conditions that could classify the wheat cultivars into tolerant and susceptible categories. He has applied the image-based stress indices on satellite imagery and developed a multivariate model for wheat yield prediction during both drought and non-drought years in the state of Rajasthan. Out of his research work, he has published 2 papers in NAS-rated journal Ecological Indicator with a NAS impact factor of 10.96. The next PhD student, Mr. Bhavani Prasad Mandal, has worked on digital soil mapping using imaging spectroscopy. His study region was part of Katol block in Nagpur. For his work, he has acquired 4 different kinds of spectral data, namely lab-based Vis-NIR spectra, absorbance and emissivity of MIR, and airborne Vis-NIR spectra with Everest NG sensor. The soil organic content map prepared by him by using the kriging technique gave an R-square of 0.45. With airborne map alone, he could achieve an R-square of 0.51. However, when airborne map was combined with the environmental covariates, the resulting digital soil map prepared by him had a maximum R-square of 0.55. Digital maps of soil fertility properties were generated utilizing airborne imaging hyperspectral data of Everest-NG in combination with multispectral data and environmental covariates gave acceptable accuracy irrespective of vegetation cover for all the soil parameters. MSc student Mr. Tarun Kumar did his thesis work on artificial intelligence-based image classification for identification of seeds of wheat cultivars. He had collected more than 1,000 images of each wheat cultivars harvested during 2 seasons at 2 locations, namely IARI New Delhi and Karnal, using the photo chamber devised by him. He also implemented image augmentation of the wheat seed images by creating random changes in rotation, brightness, contrast, hue, and saturation of the images. Out of the 5 CNN models used by him, he observed Inception ResNet V2 performed the best. He implemented image augmentation and finally modified the hyperparameters and developed the CNN model for image-based identification of seeds of wheat cultivars. He observed that the CNN model Inception ResNet V2 had an accuracy of 68.3%. It was further improved to 79.4% through image augmentation, and finally through optimization of hyperparameters, an accuracy of 85.3% could be achieved to identify the seeds of the wheat cultivars from its images by using the artificial intelligence-based image classification technique. Next, MSc student Ms. Sailaja Rastogi worked on remote sensing of rice residue burning and conservation tillage in wheat. Spectroradiometric observations collected by her at each wavelength showed significant differences among the 4 different feature classes, namely mature crop, harvested crop, burnt crop, and crop harvested at the surface. Observations were also collected by her from fields with crop harvested at different heights during pre-burning and post-burning scenarios. In the crop fields with different heights of stubbles during harvest, no significant differences were observed in the signature of the burnt crop. Similarly, among the different multispectral vegetation indices evaluated by her, she observed that the indices Carey and NBR were the best to characterize the mature and burnt crop respectively in the field. She also developed the rice classification and burnt area map from Sentinel-2A image dataset. Similarly, regional map on conservation agriculture practices in the districts of Karnal and Kaital were prepared by her and their statistics could be derived. Next, MSc student Vibhuti Bhushan Shetty worked on mapping of rice crop using microwave and optical remote sensing data. The student has used both microwave and optical remote sensing data from satellites Sentinel-1 and 2 respectively. Spectral response curve from both VV and VH polarization using multi-temporal C-band microwave satellite data showed significant differences under VV polarization for characterizing different crops like rice, cotton, jowar, etc. He generated rice area maps from both microwave and optical remote sensing data for different districts of Punjab and Haryana states. He compared the satellite-derived rice area with state-reported data and observed less deviation with microwave satellite data than optical data. The next MSc student, Mr. Abradip Sarkar, worked on the evaluation and prediction of structural stability and hydraulic conductivity in saline soils using machine learning. For his thesis work, he collected soil samples from Nilokeri, Nisang, and Asan blocks of Karnal district. He developed spatial maps of different soil parameters. Following machine learning approach, he predicted soil structural stability and hydraulic conductivity using easily measurable soil parameters. For the prediction of hydraulic conductivity, he observed high correlation coefficients using support vector machine and low RMSE was observed in case of multilinear regression for texture data and support vector machine for fractal data. However, for the prediction of mean weight diameter, support vector machine was observed to be the best. The next MSc student, Krishna Kumar Sudhanshu, worked on simulation of soil profile moisture and water productivity of mustard cultivars using aquacrop model. He had conducted an experiment with 3 mustard cultivars under 2 different irrigation during normal and late zone conditions. He had calibrated and validated the aquacrop model for predicting biomass, soil moisture profile, and yield of mustard crop with an error of around 4 to 10%. Through simulation exercises, he observed that during a wet year like 2012-13, only 1 irrigation is sufficient for mustard cultivars to have increased seed yield, biomass, and water productivity. However, during a dry year like 2017-18, if sufficient water is available, then 2 irrigations are recommended to increase the yield. If water availability is less, then only 1 irrigation is recommended as it increases the water productivity sufficiently. During the last year, our faculty have conducted training programs on modeling soil processes, geoinformatics in agriculture, and 4 programs in collaboration with ISRO. During the year 2021, the students of the discipline of agricultural physics have received 4 Best Poster Presentation Awards in national and international seminars. They have also received awards in Agri Hackathon. Apart from academics, our students have actively participated in the annual cultural and sports meet and have won many prizes. Coming to students' placement, our PhD degree recipient, Dr. Kaushik Banerjee, has joined as ARS scientist. Dr. Bhavani Prasad Mandal has joined as assistant professor. Our MSc degree recipients have joined back in IRI in the PhD program. I am happy to share that during the last year, 21 publications have come out from students' research, out of which 7 publications were in journals of NAS rating more than 10. During the year 2021, many awards were received by our faculty in the discipline of agriculture physics from various academies and societies. These include Fellow of NAS, Recognition Award, Women Researcher Award, Dr. K.G. Tejwani Award for Management of Natural Resources, Distinguished Scientist Award, and our youngest faculty received the IRA Bennett Medal for PhD during the last convocation. With this, I thank you all for your patient hearing. I acknowledge the support and contributions from our student, faculty, previous Professor of Agriculture Physics, Dean and Joint Director Education, Director IARI, and PG School staff. Thank you.
Thank you, madam. Thank you very much for your nice presentation. Now we will move for agronomy, and Professor T.K. Das will make presentation.
Chairman of this session, respected Director IRI, respected Dean and Joint Director Education, convener of this session, heads, professors, distinguished scientists, ladies and gentlemen, and my dear students. I welcome you all. Myself, Dr. T.K. Das, Professor, Division of Agronomy, presenting the significant educational achievement of the Division of Agronomy during 2021 and 22. Agronomy is one of the leading divisions of IRI pursuing research, teaching, and extension in the field of agriculture. Teaching program in agronomy had been started way back in 1945, and IRI associateship equivalent to MSc degree was awarded first time in 1947. It continued till 1958 when MSc and PhD program were initiated. Till date, 258 IRI associateship, 347 MSc, and 533 PhD degrees have been awarded. Presently, student-faculty ratio is 5:1 and student-guide ratio is 6:1. There are 24 scientists present in the division of which 14 are PS and 5 each are senior scientists and scientists. Students admitted during 2021-22 are 31, of which 18 are in MSc and 13 are in PhD. Among the 18 MSc students, 8 are from IRI New Delhi, 4 students each from IRI Assam and IRI Jharkhand, and 2 students from NIBSM Raipur. Besides 29 MSc students of 3rd and 4th batch of Anas to Afghanistan are receiving teaching and research guidance from the Division of Agronomy. 15 students, 11 MSc and 4 PhD will receive degree in this convocation 2022. They work on different themes like nutrient management, conservation agriculture, weed management, crop diversification, integrated farming system and irrigation management. Ganesh Patel, under the chairmanship of Dr. Y.S. Sive, studied the response of chickpea varieties to zinc fertilization. You all know that many Indian soils are deficient of zinc. As per Shukla et al. 2016, out of 1.7 lakhs Indian soil sample tested, 40% samples were found deficient of zinc. Zinc influences several biological functions in plants such as pollen fertility, reproductive development, IA biosynthesis, sucrose transportation to root nodule, as well as carbon dioxide utilization and transportation during photosynthesis. Zinc plays an important role in human nutrition, and agronomic biofortification of zinc is therefore encouraged. And therefore, this study assumed enough importance. This experiment had combination of chickpea varieties and zinc fertilization. It was found that Pusa 547 variety had lower nodule count but higher leg hemoglobin content and grain yield than Pusa 3022. 5 kg zinc per hectare application to soil led to significantly higher nodule count, but seed priming with zinc mobilizing cyanobacteria led to higher leg hemoglobin content, indicating probably higher nitrogen fixation, but 2.5 kg zinc application to soil and 0.5% zinc sulfate foliar spray gave significantly higher yield. They also studied soil microbial biomass carbon, superoxide dismutase activity, carbonic anhydrous activity, as well as dehydrogenase activity. And it was found that seed priming with zinc mobilizing cyanobacteria resulted in higher soil microbial biomass carbon and dehydrogenase activity. But the activities of superoxide dismutase and carbonic anhydrous were higher in 2.5 kg zinc application to soil plus 0.5% foliar spray. Kajal Arora, under the chairmanship of Dr. R.S. Bana, studied the effect of residue and potassium management on productivity and nutrient use efficiency of wheat. The objectives are mentioned here. The experiment had combination of residue and potassium management treatments. It was found that crop residue retention at the rate of 3 ton per hectare led to significant improvement in yield, net returns, soil microbial biomass carbon and dehydrogenase activities compared to no residue treatment. With respect to these parameters, split application of 100% recommended dose of potassium with half basal plus half top dress at 25 days was superior to other potassium treatments, indicating the importance of split application of potassium in wheat crop. Prakash Chandra Singh, under the chairmanship of Dr. S.D. Mishra, studied the performance of mustard under differential placement of fertilizer and phosphorus enriched compost. The objectives are mentioned here. The experiment had combination of conventional broadcast and placement of fertilizer and organic inorganic sources of nutrient. The organic sources included FOM and phospho-enriched rice residue compost. It was found that 40 kg recommended dose of nitrogen through phospho-enriched rice residue compost plus remaining 40 kg nitrogen and phosphorus potassium sulfur through fertilizer was found best with respect to leaf area index, silico per plant, yield and net return of mustard. Again, this treatment led to significantly higher values of protein content and protein yield, oil content and oil yield, dehydrogenase activity and soil microbial biomass carbon than most treatment except with FOM. The study once again proved that combination of organic and inorganic sources and placement of nutrients superior to conventional methods with respect to productivity, profitability and soil fertility. Santosh Kumar SR, under the chairmanship of Dr. Amal Ghosh, studied need-based nitrogen management in wheat using precision gadget for optimum crop productivity and higher nitrogen use efficiency. The objectives are mentioned here. The experiment had different sensor-based nitrogen application using Nutrient Expert and Green Seeker. It was observed that nutrient management using Nutrient Expert and Green Seeker led to higher leaf area index, wheat grain yield and parcel factor productivity of A and P and K. But state recommendation was comparable on yield and gave higher factor productivity of K due to low potassium used. The study recommended that 108 kg nitrogen, 19 kg P and 38 kg K per hectare could be the optimum doses for higher yield of wheat. The study revealed that Nutrient Expert plus Green Seeker aided nutrient management was superior, giving highest net return, production efficiency and monetary efficiency. in wheat mainly due to higher crop yield and lower cost and input used. Amlan Nath, under the chairmanship of Dr. Bijoy Punia, studied the effect of zinc fertilization and microbial inoculants on productivity of chickpea. The experiment had different zinc management treatment in combination with recommended dose of NPK and microbial inoculants. They observed that recommended dose of fertilizer plus 25 kg zinc sulfate plus mesorhizobium plus arbuscular mycorrhiza plus phosphorus solubilizing bacteria application led to significantly higher activities of dehydrogenase by 31% and alkaline phosphatase by 58% and gave higher grain yield and net income. Again, RDA plus meso plus AM plus PSB with and without 25 kg zinc sulfate per hectare led to comparable but higher root biomass and nodule count of chickpea, indicating the role of microbial inoculants on root growth and nodule formation. Sudarshan S, under the guidance of Dr. Kapila Shekhawat, studied nitrogen management options for different crop establishment method in wheat under rice wheat cropping system. The experiment had combination of crop establishment method and nitrogen management option. They use LCC based nitrogen application compared with modified nitrogen application and recommended dose of nitrogen. They found that among the crop establishment method, zero tillage with residue, and among the nitrogen management option, LCC guided nitrogen application led to significantly higher soil microbial biomass carbon and fluorescein diacetate activity than rest of the treatment. Again, zero tillage plus residue and LCC guided nitrogen application resulted in highest values of year-bearing tiller, grain yield and harvest index and were superior to other treatment. LCC guided nitrogen application was also superior to recommended dose of nitrogen or modified nitrogen application. Ashok C, under the chairmanship of Dr. Rajbir Sharma, studied integrated weed management in Indian master. The experiment had combination of herbicide, crop residue and manual weeding compared with unweeded control and weed-free control. They observed that integrated weed management using herbicide, either pendimethalin or oxidazole, when combined with hand weeding at 30 days, resulted in lower yield loss by 12 to 16%, higher crop productivity and profitability. Pendimethalin was more effective than oxidazole, and imazethapine led to 100% phytotoxicity to mustard crop. Avnish Kumar, under the chairmanship of Dr. Raj Singh, studied the effect of land configuration and weed management on growth and yield of pigeon pea under semi-arid condition. The experiment had combination of land configurations and weed management options as treatments. They found that among the land configuration, broadbed and furrow gave highest pigeon pea yield and net return. Although yield was comparable between two hand weeding and metribuzin followed by imazethapine treatment, the net return was higher by almost rupees 3,200 in the metribuzin followed by imazethapine application. Abhishek Swain, under the guidance and supervision of Dr. Kapila Shekhawat, studied integrated weed management in wheat under conservation agriculture based maize wheat mung bean system. The experiment had combination of crop establishment method and weed management options as treatments. Brown manuring in maize followed by zero till wheat led to significantly lesser wheat and higher year-bearing tiller, grain yield and net Bs to C compared to CTW. Wheat grain yield increased by 28% under this treatment. Tank mix applications of clonazepam plus carfentrazone reduced weed interference significantly and increased year-bearing tiller, grain yield and BC ratio considerably. Anamika Burman, under the chairmanship of Dr. BK Singh, studied deficit irrigation management in mustard using drip under agri-horti system. They had different agri-horti system combined with deficit irrigation schedule as treatments. Among agri-horti system, moringa based system led to highest mustard seed yield, net return and harvest index. Among deficit irrigation schedules, 0.6 IWCPE gave higher values of these parameters compared to others. Again, moringa based agri-horti system gave higher oil content and oil yield, protein content and protein yield, water use efficiency and irrigation water use efficiency than other agri-horti system, and 0.6 IW by CPE compared to other deficit irrigation schedule led to higher values of these parameters in mustard. Shashank Patel, under the chairmanship of Dr. S.S. Rathore, studied performance of master varieties in diversified maize-based system under different production system. The experiment had combination of production systems and master varieties, and production system included organic management system, integrated crop management, conventional system, and conservation agriculture. They found that among the production system, integrated crop management led to highest seed yield, oil yield, and profitability in mustard. And among the varieties, PM26 gave maximum seed yield, oil yield, and profitability. However, PM28 was comparable on oil yield. Sunil Bandi, under the chairmanship of Dr. Y.S. Sibe, studied the effect of green manuring and sulfur fertilization on productivity and nutritional quality of basmati rice durum wheat sequence. The experiment had combination of green manure crop and sulfur fertilization as treatment. They found that basmati rice yield was comparable between Sesbania and Cotalira green manure, but durum wheat yield was significantly higher under Sesbania than Cotalira green manure. And 40 kg sulfur to both rice and wheat gave significantly higher yield of rice and wheat. Again, summer green manure with Sesbania accompanied with 40 kg sulfur per hectare to both crop led to higher quality of durum wheat grains, having higher SDS sedimentation value, crude protein, beta-carotene, and narrower nitrogen sulfur ratio. Similar improvement was also recorded in basmati rice grains. These photographs show the difference in sulfur application to rice and wheat crop. Ayesha Fatima, under the chairmanship of Dr. B.K. Singh, studied livelihood security, resource budgeting, soil health management under integrated farming system in northwest India. You know, IFS is the need of the hour, particularly for maintaining food and livelihood security of small-scale farmers of India. Therefore, this experiment assumes enough importance. This experiment was conducted on an existing IFS model. There are different enterprises combined together as treatment, and it was found that integration of different enterprises, namely crop, dairy, fishery, poultry, duckery, apiary, boundary plantation, biogas unit, vermicompost led to significantly higher system productivity, system economic efficiency, annual man-day employment, net energy gain, water productivity, monetary water use efficiency, and eco-efficiency over other enterprises. Bharatraj Meena, under the chairmanship of Dr. C.M. Poriyar, studied the precision nutrient management under conservation agriculture-based maize wheat system. The experiment had tillage and crop establishment combined with precision nutrient management as treatments. They found green seeker and SSNM based nutrient management increased system productivity by 29% and 31% with the additional application of 108 and 120 kg NPK over farmers fertilizer practice respectively. Net economic gain of rupees 41,000 and 47,000 were obtained with green seeker and SSNM based nutrient management over unbalanced application of fertilizer. Similarly, water use efficiency are improved under CA-based system and precision nutrient management. The CA-based permanent bed or zero-till plot with optimized nutrient application significantly improved and stabilized soil organic carbon and had positive impact on soil health. This also led to 16% less carbon footprint on spatial scale and could be an environmentally safe practice. And this was water as well as energy use efficient. Sunaka Ghosh, under my guidance, studied nitrogen management and weed dynamics in a conservation agriculture based maize wheat movement system. She studied in the 9th and 10th year of a long-term conservation agriculture experiment. The treatment combination were zero-till, permanent, narrow, broad, flat bed with and without residue compared with conventional tillage system. The residue plot had again 175% nitrogen. She found that permanent broadbed with residue and 100% nitrogen led to significantly higher system productivity and net return and output energy, but zero-till flatbed with residue was comparable. The permanent broadbed plus residue plus 75% nitrogen, however, gave significantly higher partial factor productivity of nitrogen due to lower rate of nitrogen used and comparable yield obtained. It was also found that CA-based permanent broadbed with residue 100% nitrogen led to higher root length, root biomass and root volume density than CT system. All CA-based permanent, narrow, broad and flat beds with residue had significantly higher ammonium and nitrogen than CT. Also, CA-based system had higher proportion of ammonium nitrogen, almost 3 times, than nitrogen, that was 1.5 times compared to CT system. Again, the CA-based permanent broad bed, narrow bed and flat bed with residue 100% nitrogen reduced weed seed bank considerably. The CA system had significantly higher soil organic carbon storage, but permanent broad bed and permanent flat bed proved superior. Permanent broad bed with residue with 100% and 75% nitrogen gave comparable system yield, indicating a saving of 75 kg nitrogen per hectare in maize wheat mung bean system. And this CA system had lower global warming potential and greenhouse gas intensity compared to CT, indicating environment friendliness of this CA system. The Division of Agronomy conducted one online training program. The degree recipient student published 11 research papers, of which 4 are in more than 7 NAS rating. All MSc students pursuing PhD and the PhD students are in job such as ARS scientist or assistant professor. Our student received best PhD thesis, best MSc thesis, best research paper, best oral and poster presentation award from different societies. Our student participated in IRI annual sports meet and received a large number of prizes. Our faculty have received several awards and recognition such as NAS Fellowship, NAS Associateship, Fellowship of several societies, Outstanding Scientist Award, Young Scientist Award. I am highly thankful and grateful to Director IRI, Dean Director Education, Joint Director Research, Joint Director Extension, scientists of Agronomy Collaborating Division, students, other staff of Agronomy and IRI for their help, cooperation and sincere advice. Finally, I thank one and all.
Thank you very much, Professor Das, for your very nice presentation as well as very good collation of the information. And we will move to next discipline, environmental sciences. Professor Naresh Kumar, S. Naresh Kumar will make presentation.
Good afternoon, everyone. I would like to welcome all you to the professor's presentation on environmental sciences discipline students' academic achievements during 2020-21. The Division of Environmental Sciences is a multidisciplinary and integrated faculty working to address the issues on environment and climate resilient agriculture. The mandate of the division includes conducting basic and strategic research, imparting postgraduate education and training, and providing advisory services. We have a total of 19 faculty members, including those from Niazim Baramati and IARI Jharkhand. So far, 91 MSc and 56 PhD students are graduated from the discipline. They are well placed in ARS, SAUs, ministries, or institutions of state or central governments, and some of them are pursuing their careers abroad. Currently, 21 MSc and 30 PhD students are on roll. Coming to the significant research achievements of postgraduate students during 2021. This year, 8 MSc students are eligible to get their degree, and they have worked on 2 thematic areas, basically on climate change and agriculture, 4 MSc students, and on environmental pollution, 4 MSc students. Coming to the first thematic area of climate change and agriculture. Student from Kenya, Pauline Kimani, has conducted her research work to quantify the interactive effect of elevated temperature and irrigation on growth and yield parameters of kidney bean, and also to calibrate and validate the kidney bean dynamic simulation model to apply for climate change impact studies in selected locations of India and Kenya. She conducted the experiment in the field and temperature gradient tunnels with a combination of 7 temperature, including growth phase wise exposure and irrigation treatments. Following that, the kidney bean model was calibrated and validated and applied for assessing the climate change impacts on yield of kidney bean in 20 locations of India and Kenya. The field experiment results indicate that the mean temperature of 24.2 degrees centigrade resulted in higher yields than when the crop was grown at lower temperatures. The temperature shocks of more than 3 degrees centigrade during pre-flowering and flowering significantly reduce seed yield, whereas the addition of water stress, where the soil available water content goes less than 10%, significantly reduce the kidney bean seed yield. Kidney bean model developed at the division was calibrated and validated and applied for the climate change studies. Results indicate a significant spatiotemporal variation impacts. For example, in Patna, seed yield is projected to reduce, while in Pune, it is projected to increase in RCP 4.5 scenario. Similarly, in Kenya also, at Embu location, yields are projected to increase in all scenarios, while at Kakamega, it is projected to decrease significantly in all scenarios, warranting adaptation. Mayank Tiwari conducted his MSc work with the objective to collect primary data about tree height and diameter at breast height and draw a comparative analysis of carbon storage among the various tree species growing in the IRI campus. He collected data on 5,368 trees belonging to 64 species and 27 families. He estimated the total biomass using the allometric equations, and estimation of total carbon stored in the tree and then CO2 equivalent stored in the tree were followed. The Schonen-Wiener index of more than 3 indicates good species diversity in the campus. Carbon stock is more in Terminia arjuna and Ficus religiosa trees. The total CO2 equivalent carbon stock in more than 5,000 trees studied in campus is estimated at more than 452 tons of CO2 equivalent. Latalmavi has conducted her MSc work to quantify the growth, development, physiological response and yield of tomato to different temperature regimes under protected cultivation and in open field, and further to dynamically simulate tomato yield variability conditions. She conducted experiments in CPCT and in open field on tomato with 7 temperature treatments. Then used the simulation model to optimize the climate conditions for greenhouse grown tomato. Results indicate that temperatures more than 38 degrees centigrade during flowering affected tomato yield. Temperatures above 34 bar 13 degrees centigrade, that is the maximum and minimum temperatures, also affected the yield. Temperature of 24 bar 10 degrees centigrade along with 500 ppm CO2 and 500 micromole per meter square par is found to be optimum for the greenhouse grown tomato. Kokila studied the impact of plant extracts and plant growth promoting microorganisms on tomato crop yield and fruit quality. She has done seed viability studies using 17 treatments in low and optimum temperatures. Best performing 6 treatments were further used for pot experiment. Results indicate that at optimum temperature, seeds treated with Bacillus subtilis had higher germination percentage, while at low temperature, those with MLE, that is Moringa leaf extract, had a higher germination percentage. In ambient conditions, application of Bacillus subtilis and Trichoderma hergianum increased the tomato yield and quality. Coming to the studies on environmental pollution theme, Pooja LR worked on reducing ammonia volatilization using organic and inorganic amendments in wheat field, and also to evaluate the nitrogen use efficiency and productivity of wheat with these amendments. She has quoted urea with the different amendments and applied to wheat. Results indicated that urea treated with N-butyl thiophosphoric triamide resulted in 40% reduction in ammonia volatilization loss as compared to the 20.4 kg per hectare per season volatilization loss from the untreated urea applied at recommended dose of fertilizers. Shamim has worked on the effect of industrial gypsum from LD slag on soil health and tomato yield. He used industrial gypsum from LD slag and mined gypsum in a total combination of 11 treatments in a field study on tomato. Results indicated that the gypsum application significantly increased organic carbon and microbial biomass carbon content of soil in the layer of 0 to 15 centimeters. More accumulation was found with the application of mined gypsum. The growth and yield of tomato was also better with the application of mined gypsum. However, certain quality parameters of tomato fruit were found to be better with the application of industrial gypsum. Ajay has studied the effect of chelating agent treated with LD slag on growth and heavy metal accumulation in spinach. He used 4 chelating agents, and after optimizing the concentrations and time for effective chelating of heavy metals in LD slag, he applied the LD slag, which is treated with chelates, to spinach. Results indicated that the chelating agents reduced heavy metal content in LD slag, with ethylene diamine and disuccinic acid being most effective one. The pot experiment also indicated that the translocation of cadmium and lead was non-detectable in the spinach leaves, whereas about 95% of cadmium and lead were found to be in the soil. Priyanka has conducted a study to develop an efficient method for microplastic analysis suitable for wide range of soil matrix, and also to assess the abundance and properties of microplastics in sludge amended farm soils. She used 6 soil matrices and 4 types of polymers for her study, and the initial characterization of microplastics was done through Fourier transform infrared spectroscopy. After confirming that, she also explored the use of visible near-infrared spectroscopy for estimation of microplastics in different soils. After initial characterization with FTIR, she has done the spectral reflectance characteristics with visual NIR spectroscopy and developed a rapid, low-cost, and reliable method using the visual NIR spectroscopy. This method can be employed for estimation of microplastic pollution level of more than 2 grams per kg of soil. During this year, we have a pleasure of inducting one of our esteemed colleagues from NVPGR, Dr. P.K. Mahapatra, as the Faculty of Environmental Sciences. During the year, our students have several awards and achievements to showcase. Our students also bagged the Best Presentation Awards and Best Article Awards, as well as the Newton Baba PhD Placement Program Award. And 7 PhD students have the UGC JRF. 1 student is having the ICCR International Fellowship. 4 students are having national fellowships, and 4 of them are having ICR JRF. Our faculty are also well recognized at the national and international level, including the prestigious organizations like World Meteorological Organization, NASA, IPCC, and also the central and state ministries, including DST and DBT. During the year, a total of 30 publications were made, 16 of which are from the students' work, with the highest impact factor of 4.13. The faculty publications have highest impact factor of 6.51. The environment science family thanked the IRI authorities, PG School, Associate Dean, Joint Director, as well as the ICR for all their encouragement and support. Thank you.
Thank you very much, Professor Naresh Kumar. Now it's turn of microbiology, and Professor Radha Prasanna will make presentation.
Director, Dean, Joint Directors, distinguished experts, my dear faculty members and dear students, I am here to present the significant educational achievements of the Division of Microbiology, which has been actively involved in the production and sale of microbial bioinoculants and expanding the valorization of microbial products. PG students, we have on rolls at present 55 students, among which 21 are MSc students and PhD students are 34. They have come from various parts of India. In the present convocation, there are 7 MSc students receiving their degrees. Their research work has focused on the major theme areas as microbe-mediated stress alleviation to sustain crop productivity, biomass valorization and processing employing microbes, plant-microbe interactions, and CN transformations. The first student, Mr. Jayasurya, who worked under the guidance of Dr. Meenakshi Grover. The thesis title was Pearl Millet Azospirillum Interactions under Moisture Deficit Conditions. They characterized several azospirillum strains for their plant beneficial traits and further took up the inoculation in pot experiment with pearl millet under moisture deficient conditions. The characterization of these strains under different levels of PEG showed higher proline concentration, good growth, and when used with seeds, they showed a higher seed vigor index. Such strains were taken up in the pot experiments, and also, although the moisture deficit stress showed a negative effect on leaf chlorophyll, the inoculation improved significantly the plant growth as well as root architecture. Higher leaf proline was recorded and higher sugar content. Better antioxidant status was observed when microbial inoculation was done both under 75% field capacity and 100% field capacity. Such azospirillum strains show promise as bioinoculants for semi-arid and arid region crops. Ms. Kavya, under the guidance of Dr. Govindsamy, worked on characterizing the PGP potential of actinobacteria for inducing tolerance to salt stress in rice. They screened and characterized actinobacterial isolates for salt stress tolerance and further evaluated the potential of such strains for rice growth promotion under the stress conditions. In this effort, they had a totally 40 culturally characterized actinobacterial isolates which had been earlier purified from exo and endorhizosphere of rice. These were screened for their salt tolerance in different levels of NaCl. Further, such strains were also characterized in terms of plant beneficial traits and taken up in Petri plate studies followed by growth tube and pot culture studies. They were able to show that under the salt stress, the actinobacterial plants showed significantly improved plant growth, RWC, as well as higher accumulation of proline and antioxidant contents, along with improved root traits as compared to the uninoculated control plants. These hold promise for as bioinoculants, not only for imparting stress tolerance, but also improving the plant growth. Ms. Kritika, under the guidance of Dr. Sangeeta, worked on characterizing and evaluating bacteria from stressed habitats for enhancing thermotolerance of mustard. They were able to screen several bacterial isolates from stressed environments and evaluate them for the alleviation of high temperature stress during early seedling growth in mustard. Among the set of 10 strains that they screened, they were able to select 3 strains. These 3 cultures exhibited high IAA, GA production, P solubilization, and ACC deaminase activity. Such cultures were inoculated in a pot experiment, which showed that although the high temperature stress drastically reduces several plant growth parameters, but microbial inoculation significantly increased these values, including plant biochemical parameters like proline, sugars, and phenolics. Interestingly, under temperature stress condition, inoculation also reduced the plant canopy temperature. The best strain was identified as Bacillus strain MRD17, which is further being taken up in field level. Under the theme 2, Ms. Sonam Priyadarshini, under the guidance of Ms. Anju Arora, worked on lignocellulitic potential of actinobacteria for their application in biomass processing. As we all know, lignocellulosic biomass is an important bioresource and its processing is preferred through biological means. Why? Because the energy requirements are less and it's cost effective and environment friendly. Several microbes are known for their involvement. So they isolated, screened and selected potent actinobacterial strains with lignocellulitic potential, and they characterized its enzymatic profile and further evaluated its biomass conversion potential using rice straw as substrate. 13 actinobacterial strains were isolated and purified from leaf litter samples. The enzyme assays were done, and 2 potent strains identified as Streptomyces species were further taken up for studies. Solid state fermentation was carried out using the 2 isolates with paddy straw as substrate at 40 degrees, and they found higher enzymatic activities, and structural analysis of these solids showed that the alkali washed inoculated solids had higher irregularities, which illustrated greater degradation. This was ably supported by the XRD and the FTIR data. The compositional analysis showed more lignin degradation and greater availability of cellulosic residues, which led to higher sugar release in the buffer extracts. Further focus is towards achieving higher saccharification efficiency and more sugar recovery using these thermophilic actinobacteria. Mr. Pulkit Chawla, under the guidance of Dr. Rohan Tiwari, worked on optimizing a protocol for the enhanced production of phycobilly from a selected cyanobacterial strain, Nostoc strain. Cyanobacteria, as we all know, are a very rich source of pigments, and these pigments serve not only as nutraceuticals, pharmaceuticals, but an excellent source of colors. So they screened a set of 10 Nostoc strains, and the selected strain was further used for devising a protocol for enhancing its production through modulation of various factors such as light quality, nutrients, pH, and they found that the pH of 8 was most suitable, and they developed a medium with the appropriate concentrations of various nutrients which enhanced phycobilly production. This needs to be scaled up. Under the theme 3, Plant Microbe Interaction and CN Transformation, Mr. Ashwani Dhimra worked on the influence of genotype on root nodule morphology and function in chickpea under the guidance of Dr. K. Swarnalakshmi. They characterized a set of Mesorhizobium strains for their plant beneficial traits and also studied the temporal variation in nodule development in chickpea cultivars and deciphering the relationship between nodule morphology, function, and plant growth. This is very essential as several times there is an absence of suitable strains and there is a poor rhizobial survival in the soil. These strains that they took up were characterized for the various plant beneficial traits like pea solubilization and seed germination assays, IAA production, and further they were inoculated in pot experiment which showed that increased number of nodules in the lateral roots at various crop stages with Mesorhizobium inoculation. Light microscopy showed that the nodules showed the Mesorhizobium and which perhaps had entered through the root hair curling, and V-shaped bacteroids were also observed in the nodule thin sections. Interestingly, the nodule fresh weight was high in Mesorhizobium inoculated in BG372, while it was higher at the flower initiation stage in BG3022. N uptake and plant dry weight in these genotypes was significantly improved with Mesorhizobial inoculation, and the nodule fresh weight was found to be positively correlated with N uptake and plant weight. These are being further taken up at field level. Mr. Manoj Benpadi, under the guidance of Dr. Geeta Singh, worked on the role of microbial glycoproteins for carbon sequestration in agricultural soil. Although the microbial contribution to soil carbon is well known, however, there is little understanding regarding the microbial community dynamics and the metabolites which facilitate aggregate formation and stabilization. So towards this, they took soil samples from organically cultivated fields as well as the conservation agriculture base of fields. They identified the saprophytic and symbiotic fungal component of the native microflora and further tried to understand the signature biomolecules which are involved in the seed sequestration. Their studies were undertaken with samples from fields which had zero till or they were conventionally tilled, and also with different biofertilizers, vermicompost, FIM, and very interesting results were shown in terms of the microbial communities as well as the carbon pools. The microbial community in the zero-tilled was found to be fungal dominated, as revealed by higher fungal metabolites and components just as glomalins, melanins, chitins, which show a high homology to the recalcitrant seed pools. On the other hand, the conventional tilled rice and wheat fields were found to be dominated by aerobic microbial population, and which accounted for the higher activity of oxidative enzyme resulting in loss of C as carbon dioxide. They advocate that zero-till soils may aid in the development of microbial community and their metabolites, which are known precursors of recalcitrant C. I'm ably supported by faculty members, 17 in number, along with 2 members from sister institutes, NIBSM Raipur and IRI Jharkhand. Our faculty has been extremely prolific and published 42 papers with NAS score more than 6, out of which 14 are from student thesis. They have also been very active in popular articles and book chapters in English and Hindi. Several of our scientists have won several awards and served as experts in various national panels and been invited speakers in international webinars and conferences. The division has also been involved in several collaboration and linkages for enriching the PG activities with Uttarakhand Technical University, the CSIR Academy, PAU, and also our scientists are involved in AICRP, Chickpea, AICPRP, Pearl Millet, ICR Amas Network, and many of our scientists are involved in the organic farming, natural farming programs of this institute. Our scientists are extremely active in the training of the faculty from different universities, officials, researchers, and students. Dr. Livleen Shukla in particular has been actively involved in several training programs for the Pusa decomposer application. Dr. Archana Suman, who is our biofertilizer in charge, has been very active in the production of carrier and liquid-based biofertilizers and their promotion through various workshops and training programs. Also involved in the use of microbial tools for in various studies for in the different workshops. Dr. G. Abraham has been actively involved in the application of azolla and cyanobacterial biofertilizers in organic farming and promoting their use,
ঠিক আছে
and also
আর জিডিপি
teaching
কত
the
এগ্রিকালচারের
scientists and
এরই
researchers as well as various officials in the production technologies. All our scientists were very actively involved in attending various training programs, seminars, workshops online, and presented posters as well as had oral presentations, and also attended several faculty development programs. One of our students, Mr. Mayur Naitam, presented posters in 2 international conferences and also received Young Microbiologist Award. Ms. Kritika Patil also presented a poster in the Agricultural Science Congress. 2 of our students, Mr. Hoj Hania, he was the first prize in the football winner, and as well as Mr. Yadesh, who was runner-up in volleyball during the IRI Sports Week. Several students have got gainful employment. Ms. Shruti, who completed a PhD last year, was selected as research trainee in United Phosphorus. Ms. Lavanya, serving as a scientist in ICR Institute. Mr. Pulkit Chawla, Ms. Kritika, Mr. Dipankar were selected as agricultural field officer in Bank of India. And also Ms. Shreya Birmani was selected as Assistant General Manager in Food Corporation of India. I'm thankful to the Dean, Joint Directors, as well as particularly to Dr. Manjaya and the staff of the Postgraduate School for all their support. I'm extremely thankful to our head, all my faculty colleagues and students and staff in various projects. A particular note of thanks to the advisory committee members of the various students and the facilities extended therein, the experts for their timely thesis evaluation. The National Phytotron Authority, Facility Authorities deserve a special mention as most of our experiments are taken up there. I'm also thankful to the technical, administrative and supporting staff members of the Division of Microbiology and CSUBG.
Yeah, thank you very much, Professor Radhaprasanna. Now I will make presentation on behalf of Soil Science and Agricultural Chemistry. Honorable Chairman of the session, esteemed scientists, dear students, ladies and gentlemen. It is indeed my proud privilege to present some of the most important educational achievements of the discipline of Soil Science and Agricultural Chemistry. This year, in all, 12 MSc and 3 PhD students are going to get their degree, and their research work could be classified under 5 themes. First one is carbon sequestration potential of soil. Second is assessment of soil quality. Third one is related to development of novel fertilizer product. Fourth one is enhancing phosphorus and potassium supply capacity of soil. Last one is related to remediation and assessment of metal and metalloid polluted soil. Coming to the first theme, that is soil organic carbon. As you know, under tropical condition, it is difficult to increase the soil organic carbon content to an appreciable level. Not that under such condition, we will go on adding organic matter or organic manure year after year, and there will be concomitant increase in soil organic carbon content. Rather, added organic material will be subjected to the oxidation and will be lost to the atmosphere as carbon dioxide. Hence, for effective management of carbon in such condition, we need to know the carbon carrying capacity and carbon saturation deficit of soils. Krishna Kumar Moria, under the chairmanship of Dr. T.J. Purakayastha, assessed both of these important parameters of insecticide under long-term experiment, where he found that maximum carbon carrying capacity is associated with the soil of Kalyani, followed by Kanpur and Varanasi. This is attributed to the mineralogical makeup of soil and local climatic condition. Further, using the carbon saturation deficit model, they could successfully predict the change of soil organic carbon over the year based on annual average estimated return of carbon. In late 70s in our country, it was conclusively established that under conventional agriculture, there is not much scope of carbon sequestration. But the carbon sequestration potential under conservation agriculture is yet to be examined thoroughly. Hence, Ritambara, under the chairmanship of Dr. Sheila Das, assessed the rate of carbon mineralization as well as temperature sensitivity in terms of Q10 value under conservation agriculture. Result indicated that rate of carbon mineralization as well as temperature sensitivity of the carbon is much less under conservation agricultural practices, particularly in zero tillage with residue retention as compared to conventional tillage without residue retention. It implies that under conservation agriculture, accumulated carbon is much more stable as compared to conventional practices. Biochar has generally been used as a means of carbon sequestration. Of late, efforts are being made to impart the specific property in biochar in order to enhance its utility, and such modified products are known as engineered biochar. Saptaparnide, under the chairmanship of Dr. T.J. Purakayastha, prepared engineered biochar using rice straw and treating with acid, alkali, salt solution and ozone. FTIR analysis indicate the presence of different functional groups, which in turn enhance the anion exchange as well as cation exchange capacity of biochar, and such biochar proved to be very effective in retaining nitrogen, phosphorus and potassium even in light texture soil as compared to compost. Soil quality index enables comparison of soil quality under different land uses and management practices, which could easily be conveyed to the stakeholders including policymakers. In view of limited information, Parthasarathi Gorai, under the supervision of Dr. Sunanda Biswas, assessed the soil quality under rice master system in eastern region of our country. Key indicators were worked out and higher value of soil quality index was obtained under 50% RDF plus FYM, which is also reflected in yield of the crop. Although soil quality and soil health are interchangeably used in some literature, soil health is more representative of ecological attributes of soil, such as soil biota, biodiversity, and its food web. Sujit Das worked out the key indicator of biological soil health under conservation agriculture, which include beta-glucosidase, bacterial MOEE, archaeal 16S, and bacteriated 16S. These are actually related to nutrient cycling and other important process of the soil. The value of biological soil health indicator was highest under 000 tillage with residue retention. Coming to the third theme, that is development of novel fertilizer products. As we know, in India, annual fertilizer nitrogen consumption is about 18 million ton, and you will be surprised to know that about 11 million tons are lost to groundwater, water bodies and atmosphere due to its low use efficiency. Hence, Sharat Kothari, under the supervision of Kapil Chobe, synthesized urea-loaded nanoclay polymer composite, nanoclay biopolymer composite and coated urea. Column study indicates that much lower amount of nitrogen least in case of newly synthesized product as compared to that of urea, and higher efficacy of newly synthesized product was also evident in terms of yield and nitrogen uptake by maize. In the similar line, Ravi Sahani, under the chairmanship of Dr. K.M. Manjaya, undertook detailed study on assessing the impact of urea-loaded NCPC and NCBPC on nitrogen dynamics in soil, as well as important phenotypic attributes of rice plants using the phenomic facility of our institute. Further, FTIR spectra of newly synthesized product indicate the hydrogen bonding between the OH group of bentonite and OH and NH2 groups of polymer. XRD also indicated that bentonite is evenly distributed in polymer network. Coming to the nutrient use efficiency, Sharna was the most efficient user of applied nitrogen, whereas least nitrogen use efficiency was associated with Nagina 22. Use efficiency of applied P is less than half of the nitrogen, owing to its fixation in soil due to its high affinity for different soil constituents. Hence, Sunil BA studied the distribution of phosphorus in different soil fraction in 5-decade-old long-term experiment. Results indicated that major share of applied P was accumulated in residual calcium, aluminum, and iron bound fractions, which are non-labile in nature, and this could explain the reason for low use efficiency of phosphorus in such soil. Further, regression studies indicated that not only water extractable phosphorus but also sodium bicarbonate extractable inorganic P, sodium bicarbonate extractable organic P, and NaOH extractable organic P positively contributed to the yield and P uptake by the wheat. Such basic information should be very useful in devising the meaningful strategy for enhancing the P use efficiency in such soil. Dabrup Ghosh, under the chairmanship of Dr. Mandira Barman, assessed the impact of applied silica on available phosphorus in soil because activity of silicate and phosphate ions in soil solution is governed by common solid phases in soil. Results indicated that due to silica application, solubility as well as availability of phosphorus in soil was enhanced, and the impact of such enhanced availability of phosphorus was also reflected on the yield and P uptake by wheat crop. Compared to conventional agriculture, limited information is available on dynamics of phosphorus under conservation agriculture. Hence, Chinmoy Behera, under the supervision of Siladas, studied the solid solution equilibria as well as solid phase speciation of phosphorus in soils under conservation agriculture. Results indicated that Langmuir adsorption maxima as well as affinity coefficient of phosphorus in soil was reduced due to zero tillage with residue retention, and under same treatment phosphorus was also mobilized from relatively non-labile pool to labile pool. This indicates that P management protocol specific to conservation agriculture has to be developed for proper management of phosphorus under conservation agriculture. We are totally dependent on import for potassic fertilizers in spite of the fact that we have largest deposit of mica, but as such mica or waste mica cannot be used in agriculture as a source of potassium due to its poor solubility. Hence, Khushbu Rani, under the supervision of Dr. D.R. Biswas, isolated bacteria from mica mining area, and out of those bacteria 2 strains of Acinetobacter species were proved to be efficient solubilizer of potassium from mica. Evidence of dissolution of some portion of mica at the edges was also observed through scanning electron microscope. Further, pot culture study indicated that application of mica with K solubilizer and rice residue has potential to improve the available K status of soil as well as K uptake by wheat crop. Coming to the last theme, As we know, assessment of heavy metals in soil is tedious and cumbersome using the conventional method. Therefore, Chinmoy Rai, under the chairmanship of Dr. Nayan Ahmed and in collaboration with Dr. R.N. Sahu of Agriculture Physics Division, assessed the feasibility of using visible and near infrared reflectance spectroscopy in assessing the metal pollution in soil. For this purpose, spectral reflectance data were generated for 136 soil samples, and based on this data as well as metal content of this soil, several linear and nonlinear models were developed. Results indicated that support vector machine was more efficient in predicting the metal content in soil as compared to artificial neural network as well as partial least square regression. However, further work needs to be carried out in this direction. Elevated levels of arsenic in groundwater have been reported in some places of Jharkhand. We know the mobility, bioavailability, as well as toxicity of arsenic depends on solid solution equilibrium of arsenic in soil. Hence, Deepak, under the supervision of Dr. Ruma Das, studied the arsenic adsorption and desorption behavior in two soil groups, that is alfisol and inseptisol. Results indicated that arsenic adsorption in soil is exothermic in nature, and more of arsenic adsorbed at low pH as compared to that of high pH. And soil, in soil, oxides as well as clay humus complexes are two important sites of arsenic adsorption. Such basic information will be very useful in devising the effective remedial measure of arsenic in soil. In the similar line, Sierra Mina, under the supervision of Kapil Chobe, assess the impact of functionalized clays on immobilization of arsenic in soil. For this purpose, a series of functionalized clays were prepared through organic and inorganic treatment. Results indicated that modification of kaolinite, bentonite, and red mud resulted in a changes of interlayer space. Incorporation of functional groups led to the enhanced arsenic sorption by increasing surface area. Among the modified clays, iron bentonite, DM, SO bentonite, and red mud were found to be most effective for reduction of arsenic uptake by different plant parts. Low productivity of acid soil is mainly attributed to low base saturation, aluminum toxicity, as well as impaired microbial activity. Hence, Subharanjan Nayak, under the chairmanship of Dr. Sarvindra Kumar, studied the aluminum dynamics and microbial function in an acidic inseptisol under long-term organic rice cultivation. Results indicated that due to application of enriched compost, aluminum was redistributed from labile to relatively non-labile pool, and same is reflected in aluminum concentration in different plant parts under organic rice cultivation. And there was also improvement in enzymatic activity as well as microbial population under organic practices. Hence, application of organics may be an effective option in acid soil to get rid of aluminum toxicity problems. We had one ICR postdoctoral fellow, Dr. A. Manikandan, who worked on development of biochar nutrient composite for cotton growing in calcareous soil. So this is all about research contribution of our student. We have 21 faculty members and we could publish 41 research papers. Out of 41 research papers, 21 came from student thesis. I am very happy to say that out of 41 papers, 13 paper we could publish in journal having NAS rating more than 10 and 3 paper having NAS rating more than 15. Our student also excelled in various fields. Master Kiran Kaur got merit medal and Dr. S.P. Raichodhuri gold medal. Prem Kumar, he got ISSS Zonal Award. Mr. Sharat Kothari, he also got Dr. S.P. Raichodhuri gold medal. Mr. Anit Das, Mr. Khurshid Alam and Vishwanath Chauhan, they got recognition in oral and poster presentation of Saloni Tripathi became overall girls sports champion. Nidhi Luthra, Amit Kumar Das, and Surya Prakash Yadav also did well in extracurricular activity as well as. Coming to our faculty, Dr. D.R. Biswas inducted as Fellow of NAS. Dr. T.J. Pudakayastha became Fellow of Indian Society of Soil Science. Devashish Goloi under Fulbright Fellowship, he is on deputation. 5 of our students started their professional career in prestigious department, organization, and institute. We could conduct 4 national level training during the period under report. I am highly grateful to Honorable Director, Joint Director Education, Joint Director Research, Joint Director Extension, and our own head for aptly supporting the academic activity of the division. And I am really indebted to our faculty members and students for their significant contribution in educational achievements of the division. That's all. Thank you. Thank you very much. So now we will take up the last presentation of this session, and Professor Mansingh will make presentation on behalf of Water Science and Technology.
Chairman, sir, with your permission, I am presenting the significant academic achievements of the students and faculty of Water Science and Technology for year 2021. We have faculty strength about 11 and 36 students. We train the students in a multidisciplinary mode with an expectation that in their future career they would always become a part of a multidisciplinary team and can contribute. This year, 2 students, Aditya and Vignesh, completed their program under the supervision of Dr. D.S. Gujar. Their research work was in the theme area of wastewater utilization and management. Aditya assessed the impact of wastewater irrigation in marigold crop. The focus was wastewater irrigation scheduling criteria. He chose 3 objectives, 8 treatments. The focus was the irrigation scheduling criteria. In maximum allowed deficit or management allowed deficit, he used the depth of irrigation as one of the criteria, bringing the moisture at field capacity. This is a view of his experimental area. The results he found that 50% management allowed deficit was the best irrigation scheduling criteria. Wastewater irrigation had significantly enhanced the flower size, yield and net return as compared to the groundwater irrigation. Vignesh attempted heavy metal spiked wastewater irrigation impact on soil health, productivity and metal translocation in marigold. His objective was estimation of heavy metal translocation in marigold by measuring bioaccumulation factor, translocation factor, and he attempted a sort of whether the marigold has a potential for phytoremediation of heavy metals. This is his experimental view. Aditya worked in the field scale, he worked on a pot experiment, but both the students were very industrious and hardworking, and their finding was here like marigold may be chosen for the phytoremediation of heavy metals from metal contaminated soil. He also put a rider that a long-term monitoring on impact of heavy metals rich wastewater irrigation in marigold needs to be studied more in different locations. This year, the publication of the journal publication from the students and faculty reduced to 15 as compared to the last year 24. This was attributed to the reason of pandemic. In this year in water science technology, 6 PhD students, they were supposed to submit the thesis, but they got delayed because of uncertainty of traveling from institute to the home and so on. This is the reason that the publication was less this year. Some noteworthy interjection in national debate. On 1st April 2021, I wrote an article in editorial. The title was Dusri Harit Krant Ki Roop Rekha. This got published on the editorial page. Then in November, Sanjay Gupta wrote an article on Pradushan Rokne Ke Aadhe Adhure Upay. On that also, I wrote an interjection, and it was like Samagrata Mein Ho Vikas, and this was also published on editorial page. Similarly, very recently, on the natural farming theme, an article appeared from Vice Chairman Niti Aayog and Neelam Patel, Kheti ke taur tarikon mein badlav ka waqt. On this also I wrote a comment, and that was published. Then the comment here is समय की आवश्यकता है प्राकृतिक खेती. In the budget also, there was announcement that there would be natural farming all along 5 kilometer wide towards the Ganges. Recognition and awards. I am associated with certain committees where I am contributing professionally. Noteworthy are Wetland Authority of NCT Delhi and Equipment Purchase Committee of IRI that takes considerable time in clearing the files. Dr. Ravinder Kaur, also chosen as Associate Editor for Soil Health, Technical Expert in the VIRAX Committee. Steering Committee, she was nominated by the DG for the Ministry of Jal Shakti for the formulation of sewage sludge. Dr. Manoj, Secretary General of ISA. Similarly, Sushma Sudhasi also recognized from her, Women's Achiever of the Odisha, Best Scientific Award, Editorial Board, and so on. Neeta Dwivedi was also recognized as a Fellow of the Society of Tropical Agriculture. Dr. Bipin Kumar is a Chief Editor of Pusa Samachar, which is a popular IRI YouTube channel these days. Students are very active here, particularly the girls. Neha Singhal participated in a training workshop. Saujanya bagged the Best MSc Thesis Award from the 5th International Agronomy Congress. Aditya also bagged second prize in poster presentation. Dina Dhal, Kirtuga, they are also participated in academic activity. This is another display of a teamwork of the faculty and the students. They have portrayed a live model in the Krishi Vigyan Mela for the Water Secure Atmanirbhar Bharat. Also, we celebrated World Water Day on the guidance of Director Dr. Aki Singh. Dr. M.K. Sinha delivered a talk on valuing water prerequisite for achieving Sustainable Development Goal. Also, we receive a lot of important visitors from the ministry. On the December 1st, we received the Additional Secretary and Joint Secretary, Ministry of Agriculture. They mentioned very categorically that they have come here to learn about pressurized irrigation. So our team did interact with them for an hour. Then another visit was on 13th January by another new Joint Secretary who was inducted laterally in the Government of India. He was very keen and also alumnus of IARI. Then why HRD in water science technology is so critical and important. I've written an article, Atmanirbhar Krishi Ki Kunji Hai Sinchai, November 17, 2021. And if one reads this article at full length, one would very easily understand why the HRD in water science technology is very, very critical and important for the country. Finally, I acknowledge for the support and cooperation from the colleagues, particularly the faculty of water science technology and other discipline of School of NRM, all the students of water science technology, the dean, associate dean, and colleagues from PG school, finally, Director ICR IARI for their support. Thank you so much.
Thank you very much, sir, Professor Mansingh, for your very nice presentation. Now, Chairman, sir, we could complete all 7 presentations. And now I invite you for your kind remarks and comments. Thank you, sir.
Thank you, Shiva. Thank you very much. It was a pleasure to listen to you all. And I particularly appreciate this fact that everything was pre-recorded so that time management is not a problem for the chairman or the convener. It's wonderful. I liked it. All the presentations, all the divisional presentations were wonderful. And the student research was as per the requirement of the time in most of the cases. And everything, all the experiments and all the projects were laid out systematically so that the students get the initial idea of experimentation itself, number 1. Number 2, selection of topics. And number 3, the correct methodology. And also subsequent parts of the statistical analysis and all those, they must have taken care by individual guides, PhD guide or advisors. But a few points I would like to make here. In all the discipline, although the topics, choice of topic was as per the demand of the time, but still there is a scope to explore the new dimensions which are happening and new contribution in literature which is happening from the countries abroad, also in India. And we need to learn some new techniques also and incorporate them in our methodology so that it becomes more robust. Although in present shape, it is wonderful. In first session of agriculture engineering, I'm very happy that most of the students, they have been given the topics which are very, very relevant in present day context, particularly the sensor-based techniques, the transformation of resource conservation technology, then climate change impacts on water footprint and all. This kind of training to the students will certainly pave a path for future Indian agriculture, and also not only groundwater modeling, but groundwater quality was also given due weightage in this, its assessment. Similarly, the IoT-based solutions for aeroponics. This is also one topic which has attracted the attention. And going to the solar power, the control cooling system, and particularly in post-harvest area, the LED-based photodynamics in activation. These are some areas which are very, very important. And I believe this will show a new path to the students who have taken this research, and ultimately they will be joining some university or ICR institute. So those institutes will be benefited in future. Agriculture physics also, the crop simulation modeling, the thermal imaging, and also digital soil mapping, then AI-based some solutions in image classification models, the multispectral image classification models, also use of microwave and optical data, and the structural stability, the machine learning is used in structural stability, particularly in saline soils, where structural stability is a very, very important aspect. And also use of aquacrop and all these kind of tools in any test crop they have taken mustard. But definitely this kind of studies will, this kind of studies are going to provide very good platforms to the students in their future. The agronomical studies, although they were the kind of typical agronomic work, but they're also, what I understand, we need to explore the literature in depths and dimensions so that some new ideas can be inculcated in IRI research programs, so that students be given some new kind of work other than the kind of soil fertility experimentation. In environmental sciences, both in contamination side and climate change modeling and processes side, both the sides, the work presented here was very, very impressive, whether it was irrigation versus elevated temperature, right? The whole impact on carbon storage in standing trees, that was also a wonderful work presented here. And the ammonia volatilization, although it is a very classical and old kind of work which is already conducted in several countries, but certainly this also requires some new dimensions and some new observations have placed forward. Heavy metal contamination, the chelates and their use, and microplastics, these are the new things which are giving really new dimensions to the divisions reflections at the ground level. The microbiology, very, very impressive. Your past achievements like Pusa decomposer and several biofertilizer formulation. Besides that, still you are working on microbe-mediated the stress alleviation, also PGP potential of actinobacteria for salt stress, and also within bacteria, the thermotolerance in mustard is also wonderful work, and lignosyllatic potential of actinobacterium, phycobiliprotein production, the protocols which you have developed, and lycoprotein for carbon sequestration studies. These are some very, very impressive works, although in one or two slides it's very difficult to grasp all the, but still the first glance, what I could understand, these are wonderful things which are coming forward, and our students, definitely they are going to be a good research leaders tomorrow, and research scientists also initially. The soil science and agriculture chemistry, Shiva presented work of all the students, 12 from MSc and 3 from PhD. And that includes, besides nutrient transformation in different management practices like conservation agriculture or somewhere else in precision agriculture also, and also not only the dynamics, but some potassium solubilizers. This is yet another area, because if we devise some techniques wherein we could reduce the consumption of 15 or 10 to 15% of phosphorus and potassium fertilizers, that will be a huge saving for the country. So all the attempts and all the efforts which we are making for this, that will certainly lead to enhancement of few percentage of use efficiency of few percentage of total consumption. That will be huge saving for them. And in some cases, I have seen there is a very high scope, and particularly the use of NCPCs, although we did not come up with any practical solution or final tool or final product for this, but certainly this product has a great potential. Biochar, the industrial biochar and nutrient retention, this is yet another area which has been talked everywhere, and most of the developmental department, they are very keen to adopt it. But we need to develop some scalable and practicable solutions or scalable models, or some models which young entrepreneurs, they can come and take it up. So these are some areas where students are given training, and also not only here but Jharkhand and all, you went and explored the acid soils where aluminum toxicity and iron toxicity studies are also taken. In water science and technology, of course, wastewater remediation is an important area. Phytoremediation is yet another area, and the overall kind of research by students have done by different departments is wonderful, highly impressive, and I'm really thankful to the IRI management, particularly Director Aki Singh and the whole team, for giving me an opportunity. At least this has given me an opportunity where I could glance all the activities in a single go, and I could see what kind of research areas being explored, and what are the new themes being taught to the students, and how students are groomed in all this area. And looking at this, I feel that the future NRM will be benefited from the kind of teachings and kind of research you are conducting using your students and all. So thank you very much. Thanks a lot.
Thank you, Dr. Thank you so much. That was wonderful summary of all the presentation, and I'm so grateful to you for sparing time and being with us. I'm also thankful to Dr. AP Srivastava. I saw him sitting all through, and he has been listening to all presentations. Thank you, Dr. and welcome. It's a great encouragement, presence of people like you. And Dr. Chaudhary has given some guidance in which we should plan our research. We will certainly try to, we have taken note of it, and we will try to modulate our research program in future in this direction. So I'd like to thank you once again, Dr. for accepting our invitation and being with us in spite of, I know that there was a meeting and that continued still longer, but you could make it. Thank you so much. And we have finished exactly on time. Thank you so much.
Thanks a lot.
Thank you, sir. We are at the end of this pre-lunch session, and it is a great privilege for me to express my esteemed and profound sense of gratitude to Dr. S.K. Chaudhary, DDG NRM ICAR, for agreeing to our request and sparing valuable time. Sir, you are custodian of the natural resources of the country and intimately associated with various phases of their management, maybe for last more than 10 years. So I think you all will be agree with me that we could not have better person than Dr. Chaudhary for this purpose. Sir, your critical comments and valuable suggestion will definitely help us in devising the ways and means of the sustainable management of natural resources. So thank you, sir. Thank you very much.
Dhanyavad.